So I welcome you all in the 8th day of faculty development program entitled Exploring Multidisciplinary Avenues in Pharmaceutical Education and Research. Yesterday's lectures were very knowledgeable and informative. We were having 3 lectures yesterday by Dr. Harinder Singh, Dr. Kesar Raza, and Mr. Amit Bhatia. We enlighten ourselves regarding nutraceuticals, pharmacokinetics, and basics and advancements in dynamic light scattering. So today we should move towards today's proceedings. So I would like to invite Mrs. Navpreet Kaur, Assistant Professor, for welcome address.
Good morning, everyone. As today is the 8th day of faculty development program entitled Exploring Multidisciplinary Avenue in Pharmaceutical Education and Research. Today we have 3 technical sessions. For first technical session, I would like to welcome Dr. Vandana Patralve, Professor of Pharmaceutics, Institute of Chemical Technology, Mumbai. For second technical session, we have Dr. Manjinder Singh Gill, Assistant Professor, Process Chemistry, Department of Pharmaceutical Technology, Naipur, Mohali. And for third technical session, we have with us Mrs. Kavita Kanoje, Intellectual Property Professional, Co-founder and Designated Partner at Paths to LLP Pune. I cherish the auspicious presence of Professor Monica Gulati, Registrar and Executive Dean at School of Pharmaceutical Sciences, LPU. I also extend my warm welcome to Dr. V.K. Kapoor, Professor Adjunct GIG Khalsa College of Pharmacy, Gurusar Sudar. I would like to welcome our worthy principal, Professor Dr. Satvinder Kaur, GIG Khalsa College of Pharmacy, Gurusar Sudar. I also welcome all the delegates and faculty members. So now we are going to start our session, and I hand over the mic to Dr. Anuja for further proceedings. Thank you.
Thank you, Navpreet. So we have with us Dr. Monica Gulati as guest of honor for today's session. Dr. Monica Gulati is the Registrar and Senior Dean at the School of Pharmaceutical Sciences, Lovely Professional University, India, with more than 20 years of teaching, research, and administrative experience. Her research forte is enhancing bioperformance of drug molecules, including small and large biomolecules, phytochemicals, and probiotics, metabiotics, using conventional and novel dosage forms, especially in the area of colon targeting. She holds a keen interest in the quality control parameters of delivery systems. Her group has designed novel platform technologies to overcome challenges in therapeutic delivery of different classes of phytopolypeptides and phytochemicals. She has more than 80 high impact publications and 10 book chapters to her credit, with an H index of 30 and RG score of 41.1. She has been granted 4 Indian patents while 22 have been filed and published. She is an elected member of sectional committee of the section of medical sciences for the Indian Science Congress. She is on the editorial and review board of more than 20 journals. She is on the advisory board of 4 pharma organizations as an honorary member. So I invite Dr. Gulati for formal address. Please, ma'am.
you, Anuja. And you have been following up very diligently, I think, with all the speakers. First of all, good morning, and I would like to congratulate Dr. Satwinder for carrying out, conducting such a informative and such a multidisciplinary kind of FDP. And it is a pleasure for me to have, right in front of me, I can see Dr. Vanna Patravale. She has been a role model to many of us. Dr. Patravale, it is always a pleasure, meeting you and interacting with you in any capacity. And I hope that the deliberations of today's sessions, they lead to a very good takeaway for all the attendees. Generally, what happens, we attend the conferences, we attend the FDPs, the learnings stay with us for some time. And then eventually, we all get busy with our own respective very busy lives. So the purpose somewhere I feel is that there should be some good and some strong kind of networking wherein we could, utilize the expertise of each other, which would actually be the I would say the wishful kind of conclusion of any FDP or any conference or any conclave. So I hope that since we had such multidisciplinary speakers, Dr. Satwinder and her team, they have been, able to rope in very good speakers, covering the areas of IPR, covering the areas of phytochemistry, the analytical chemistry, formulation aspects, you name it, and she has it. So almost the entire spectrum has been covered by their team. So I hope that some good networking comes out of it, and people are able to improve their research, and rather than improving, I would say facilitate each other with the research work. So with that, I wish the best of deliberations and the best of learnings for all the attendees, for all the delegates. Thank you, Dr. Satwinder, for inviting me once again, and thank you to the entire team.
Thank
I
you
wish
so much.
the best on the, I think today is the concluding day or the penultimate day.
It's on 20th.
Yeah.
It's on
So
20th.
it is,
Please be there on 20th also. We'll be sending you formal invitation also.
Surely, surely. It is always a pleasure.
Yeah, thank you so much. Yeah, rather today, I think today's sessions serve the purpose because out of the 6 speakers, 5 are women. So dedicated to women forum, I must say. Otherwise, there is like good speaker is a good speaker, though it is men or women. I didn't like focus on that part. But by chance, today's session is women dominated. So let us dedicate this one to the Women Forum of APTI. And I'm so happy to have you with us. That serves our purpose and makes it successful and meaningful when we have speakers like you and like the speakers of today and every day, rather.
Yeah.
So I'm thankful to everyone for accepting our invitation and making it a success. It's all because of the learned and the stalwarts of pharmacy who have been addressing and giving their expert talk to the attendees. And I'm so happy to have Dr. Vandana also today with us. Though I have interacted with her in our women, pharmacist women power group, but have never got a chance to be face to face with her. So this is a lucky thing. I'll be interacting with her. And thank you, Dr. Kavita. Dr. Manjinder, I think hasn't joined yet. He'll be the second speaker in today's sessions. So let us hope this FDP will serve the purpose we intended to serve. So thank you so much, everyone.
Tamanna, I think the entire Punjab is waiting for you now.
Thank you so much. And I'm also waiting to come to Punjab. You know, it's virtual is enough now.
You've got such a strong fan following in Punjab,
Next
I am
time
really
it will
blessed.
be offline, definitely.
Thank you.
Welcome,
Thank you.
ma'am. Welcome.
Thank you, Professor Satwinder.
Thank you, ma'am. Thanks a lot for your kind words. So it's time to felicitate Dr. Monica Gulati. So, ma'am, kindly accept our a token of gratitude. Thanks a lot, ma'am, for your precious time. So,
Much.
we are proceeding towards our technical sessions. So we have amongst us Dr. Vandana Patravle, convener APTI Women Forum National and Professor of Pharmaceutics, Institute of Chemical Technology, Mumbai. Dr. Vandana Patravle is currently a professor of pharmaceutics at the Institute of Chemical Technology, Mumbai, India. She has more than 30 years of teaching and research experience. She has over 200 refereed publications with over 9,688 citations with H-index 49 and 20 patents in pipeline and 2 trademark registries. She has published 2 books and 35 book chapters with international publishers. Dr. Patravale has been active in teaching, research, and service throughout her career. She is an independent director of a device manufacturing company and listed as top 2% of world scientists in pharmacy. Her areas of research include development of nanocarriers with major emphasis on infectious diseases, cancer, and neurodegenerative disorders, medical device development, nanodiagnostics, and nano she won Fellowship of India Oration Award 2020, APTI's Dr. Manju Shri Pal Best Pharmaceutical Scientist Award 2019, Shri Amrut Modi Distinguished Researcher Award 2018, OPPI Women Scientist Award 2015, Bill Melinda Gates Grant Award 2015, Best Pharmaceutical Scientist Award 2014, Vasvik Award 2013, Veneto Nanotech Award 2013, APTI Best Teacher Award 2012, Fellowship of Maharashtra Academy of Sciences 2011, and KH Gardner Distinguished Researcher Award 2009. She is Vice President, CRS Indian Chapter, Editor, CRS IC, and APTI Women Forum newsletters, and on editorial board of peer-reviewed journals. She is actively collaborating with researchers as well as industries within country and abroad, and has completed Indo-Swiss, Indo-Japan, Indo-UK projects. She is executing all major grants from Indian government focusing on product development. She has transferred about 20 uh, technologies industry, including drug-eluting stunts being marketed in more than 65 countries. So I invite Dr. Patravale to discuss with us on multidisciplinary research case studies. Please, ma'am.
Thank you so much for the kind words. And am I audible?
Yes, ma'am, you are audible.
Okay, so kindly let me know once I share my screen if it is visible.
Yes, ma'am, it's visible.
Thank you so much, and thanks to Dr. Gulati also for those kind words, and definitely to the team of GHG Khalsa College of Pharmacy, in fact, for inviting me. So I'm doubly happy, one being a speaker and one being APTI Women Convener. You know, it is always a privilege to share the knowledge which we have gained over the years with our fellow colleagues. And what better day than today to discuss about this multidisciplinary research, which we have been able not only to do the technology transfer, but now these stents, coronary stents, are being marketed in more than 65 countries. And the same company where I've been roped in as independent director. So I would start with the presentation, and of course without flashing this photo, I do not start, because this is the Institute of Chemical Technology which has given us everything. So every day when we enter these portals of excellence, we actually read on the board that you have to give before you take. And that's the message which I would like to pass on to all my fellow colleagues. See that with focused mind, if you start putting your mind towards research and give something to the society, give something to the mankind, God is great. He'll give you everything what you desire. So these are my website details and email ID in case someone wants to interact with me. I'm always free to interact, I would say. And once again, thanking GHG Khalsa College of Pharmacy and APTI Punjab State Branch for conducting this particular FTP. If you look at top 10 global causes of death, I'm sure this slide will not come as a surprise to you. Cardiovascular disease, you can see, is the world's #1 killer. Causes 31% of all global deaths. And actually, more than 75% of cardiovascular disease related deaths take place in low and middle income countries. So that means it doesn't spare anyone and doesn't spare India in particular. And now, with the incidence of diabetes growing, this incidence is also on rise. So, though we said that 17.8 million deaths every year happen from CVD, 2027 to 2030 statistics are really not very good, which you can see in this particular slide itself, that this incidence in millions is rising. So CVD deaths percentage of all deaths is also rising by 1%, and 1% for population of billions is too much. So all in all, future with respect to CVD is not very great, and therefore you need to work towards it. If you look at the price check also, cost of heart disease is about 214 to 351 billion US dollars. So this leads to how many hours of loss of productivity, and that loss really can lead to various kind of issues. So what are we proposing? We are proposing, can we tackle cardiovascular diseases with stents? And this is these stents which I'm going to talk to you about, wherein multidisciplinary approach comes in. Because stents are nothing but, say, spring-like structures, which you'll see in the following slides. But these need to be micro-machined. And there comes the role of metallurgists. There comes the role of laser scientists. There comes the role of mechanical engineers. And then when those stents are made, we as formulation scientists have to actually coat those drugs with drug and polymer combinations so that they release the moiety in the rate at which the disease can be brought under control. Sometimes the release has to be for years together, a constant amount of release for years together. And that's where your formulation scientists come in. Of course, these need to be evaluated on cell lines. So you need biotechnologists, need to be evaluated for preclinical studies, and therefore you have role of pharmacologists, so on and so forth. So the case study which I'm taking today definitely will open your eyes towards a new field, which is devices, in case you have not already started working on. And definitely there's a lot of scope of us with this particular range of devices. So what you can see here is a cross section of a vessel. In cross section of the vessel, when the plaque starts forming, you can see yellow indicates the plaque formation. It goes on increasing. It could be due to your wrong dietary habits, could be due to some disease condition itself, or could be due to lack of exercises. So when plaque starts forming, it leads to obstructed vessels. So what is generally done is you are putting a metallic stent inside, which is taken from your thigh or nowadays even from shoulder to the vessel which is blocked. And what would this stent do is it will compress the plaque against the vessel so that it will open up the lumen. So earlier bare metallic stents were used, but now we are using drug-eluting stents because they have given lot of advantages, which you'll see in the few slides starting from now. So if you look at the market forecast also, this market definitely is a growing market, growing at 5% cumulative annual growth rate. Key market findings are actually indicating that there is shortcoming of existing treatments. That means from bare metallic stents, we moved on to drug-eluting stents, but again, there is much more scope in actually working on even drug-eluting stents, be it with respect to the drug molecules, be it with respect to the stent platforms as such, wherein you need to decrease the stent thickness as much as possible. And that needs to be done because generally there are chances of restenosis happening. That means you have implanted stent, but around the stent, the growth of normal cells takes place and takes place so rapidly that again it leads to blockage. And that's where your drug comes into picture. So DES market is growing. DES stands for drug-eluting stent. So this market is definitely growing, as I have indicated, growing all over world. But definitely Asia Pacific, we need to be little more careful, and even Middle East and Africa, where a lot of cases are seen. Drugs, variety of drugs have come into picture, but sirolimus is still the leading drug. It is, sirolimus is immunosuppressant, but this is used actually to uh, decrease the cell growth, which if it happens very fast will again lead to blockage of arteries. So segment wise, you have to work on scaffold. Scaffold started initially with stainless steel. When I started working on this technology in 1997, first stent we brought to market in 2005. It was stainless steel, which is not even listed in this slide. Then it was an era of cobalt chromium. Many platforms still use cobalt chromium, platinum chromium, nitinol, biodegradable stents as such, including magnesium stents, etc. Application wise, we are talking here only of cardiovascular. But stents are not only used for cardiovascular segment, they are also used for urinary bladder, for kidney, maybe for GI tract, wherever obstruction is seen. So this market is huge. But today, I'm focusing on only percutaneous coronary intervention, that is those which are used for vascular diseases. End user wise would be used in hospitals, cannot be used by patient himself or herself, obviously needs intervention, right? So therefore, either ambulatory surgical centers or hospitals would be using it. Product wise, I've already told you earlier, there were bare metallic stents. Still in certain cases, for example, if you have carotid artery blockage, carotid artery is within here, the neck region, people prefer bare metallic than putting drug eluting, because function of that is just to open up so that the circulation to the brain increases, there's no stroke. But in other cases, you do wish to have either polymer based stents, that means drug polymer coating, or if we can work, which we are now, in fact the trials are right now going on in Netherlands with whatever we have developed with respect to nanotherapeutics, or polymer free stents. Drug categories, I've already told you, generation wise, there are first generation, fourth generation is what we are talking about now. First generation is what we had worked on then, and I'll show you a video which will clear everything about it. And region wise, no region is left. So problem is that plaque starts forming, but it is not until the plaque burst can you actually know severity of the disease. So our diagnosis also, early diagnosis is not there. If we can work on even diagnostics, to predict whether a person 10 years hence will have a plaque formation or not, that also would be great. So that is another field which is opened up for you. So unfortunately, current stage is that unless the plaque ruptures and blood clots start forming, limiting the blood flow, you are not able to diagnose this disease, and a lot of time is wasted in that. So coronary artery disease is thickening or hardening of arteries. Results because of building of plaque, as I've already shown you. If plaque formation takes place, reduced blood flow would be there, even to the heart, and then of course to the other organs. And this reduces the supply of oxygen as well as the vital nutrients to the heart wall. Leads to chest pain like angina. There could be injury to the heart muscle, which might also lead to heart attack. What are the different treatment procedures for CAD? Obviously, earlier it was a bypass surgery, which you might have heard. And maybe some of your relatives would also have undergone it. So in this case, the blocked coronary arteries are bypassed by taking a blood vessel graft from patient's own arteries. So some other artery you're taking the graft, maybe from the thigh, maybe from the hand, and then you are placing it where it is required. So you are cutting it from one place, putting it in another place, or it's an open heart surgery, has its own disadvantages. Then there is enhanced external counter pulsation, but this cannot be used in all the cases. So you can schematically see the normal blood flow in artery is very smooth. But if plaque formation takes place, definitely the blood flow decreases. So by using this particular technique, EECP, what you are doing is you are stimulating the openings, or you are actually leading to formation of small branches of blood vessels. So instead of taking the blood through this part, they'll take it from the branches to the main artery. But this can be only done for patients who have stable angina and patients who actually do not qualify stenting also. They do not qualify for bypass, they do not qualify for stenting. So only in those cases will collateral creation help. Coming to our topic, that's percutaneous coronary intervention. We do either balloon angioplasty or it is stent angioplasty. Now, in case of balloon angioplasty, as the name suggests, it's a balloon which will be passed in through your shoulder or through your thighs to the side where there is plaque formation. And then the balloon is actually pumped in air, so it will expand. So you can see in this slide that when it expands, it is compressing the plaque on both the sides. After that, it will be deflated and taken out. This will leave the artery widened and the blood flow would get improved. So this is minimally invasive, very widely used procedure. In fact, in our lab, right now we have Prime Minister Fellows working also on coated balloons. And believe me, that is the future. As far as coronary stent is concerned, if there is buildup of cholesterol, which is to a great extent, it's a hardened cholesterol, you need mechanical intervention. So balloon is not enough. So in that case, onto the balloon will be mounted this stent. What you can see as a crisscross structure is a metallic stent. And then when the balloon is inflated, the stent also opens up and compresses the plaque. Now, balloon would be taken out, deflated and taken out, but this metallic stent will remain in the place. So this expanded stent would allow for blood circulation to be as it was before the plaque set in. So stents, in other words, are tiny wire mesh tubes which treat narrowed arteries in the human body. So essentially, they are restoring the flow of blood or even other fluids, depends on where it is actually implanted. They are permanent implants. It's not that once their function is over, you are again removing them off. Over a period of time, these body cells will grow around it. So it will become a permanent part of your body. Medicated ones are referred to as drug-eluting stents, and non-medicated are referred to as bare metallic stents. So what does fabrication of DES or drug-eluting stent involve? You need to check what are the mechanical properties. You need to be aware of what are the manufacturing processes, and you should be aware of how can you customize the stents to suit different patient population. Some will have artery length 2 mm, some will have 4, some will have 6. Plaque also would vary 1 mm, 2 mm, 4 mm, 8 mm. So customization also should be possible. As far as mechanical properties are concerned, you need to look at design and geometrical aspects. So there are different designs, and continuously work is going on in this direction to make stents better and better. The company with which I'm associated, Sahajanand Medical Technologies Private Limited, has the best stent available in the world, and that's indigenously designed. But did it happen in one day? Answer is no. Now, this company was, you'll be surprised, into laser cutting of diamonds. So while they were cutting diamonds, because a Surat-based company, and you know Surat is very famous for diamonds, they thought, can this laser be used for something else? And at that time, chairman of the company, Dheeraj Bhai Kothalia, in fact, happened to visit a chemist shop and saw a person really bitterly crying. On asking, he mentioned that it is that the relative needs this procedure, but he doesn't have money. On inquiring further, he came to know that the therapy costed more than 2 lakhs. So a thought crossed his mind, can I do something for my country? Can I cut down the cost? And that's when they contacted us. It just chance that they walked into our room. You know, ICT is a huge ocean, but I took up the challenge, and with help of all the mechanical engineers which they have and the fabrication scientists they have, they didn't have a single pharmaceutical scientist. So we started work in the lab. Very slowly, we built up the design as well as we built up the materials and the coating materials. So you can see that you need to look at metallurgy, you need to look at the design part, you need to look at what kind of metals could be used. So continuous reading is the best habit. Until date, I have not lost that habit. I'm really very happy to state that at least 5 to 10 research papers, I still read it. If I don't read, I would not know what's going on in the world. And then how can I be different than the world? And you'll be glad to know the stent which we developed, indigenously developed, got the first CE mark also. And we were the first, not only in India, but world over, to do drug polymer coatings using biodegradable polymers. At that time, world over, it's not that we were first to enter into the market. There were many players like Boston Scientific, Abbott, big players, but they were using bioerodible polymer. My theory was, when they came to me, bioerodible polymers, since they will erode, they may themselves leave a small amount of polymer around which the clot can take place. So that would be harmful to the patient. So can we have something which slowly over a period of time degrades and becomes part of the body? And the idea was readily accepted by Dheeraj Bhai. And I'm really happy that he accepted and was ready to experiment, because anyway, whatever we had to do had to go through preclinical. So that confidence he had in me. We also talked to cardiologists and understood what are the problems which are associated with current stents which were marketed in 1997, and came to know before they actually reach the site. You know, the vessels are not straight line vessels, they are all tortuous paths. So to travel that tortuous path, the mechanical integrity of the coating has to be such that it doesn't give way on the while passing itself. The drug should not get released before it reaches a site. So that also helped us to design the stent in a particular way. As far as manufacturing process is concerned, there are traditional processes and we are using laser cutting. And then upcoming trends, of course, include 3D and 4D printing. This is from literature. We are also working on electrospinning technique as well as stereolithography now, but our major focus right now also is on laser cutting. The company has put a huge plant which will be I would say, the biggest plant here in Asia for stents. As far as stent customization is concerned, you need to know the place at where they are implanted. So that means you need to have radioactivity or opacity markers. They improve the visibility. So generally at both the ends of the stent, there will be a platinum or tantalum put, because you are monitoring, the interventional people are monitoring on CCTV. They should be able to see, they can't see the stent, but they can see the platinum or tantalum. So right now we are using platinum. We are also working on this to make it cheaper. Our continuous effort is to make this affordable. Obviously, under DPCO, when it came, these stents have become cheaper, but our still endeavor is to make them very, very affordable. And if we can give to the patient something which is being sold at 1.5 lakh at 15 to 20,000 of rupees, while maintaining all the quality characteristics, that is a satisfaction which we are going to get. Fortunately for us, when DPCO came in, most of the foreign players had to leave India market because they could not sustain. So that also has increased our value, both, of course, with respect to sales also. As far as DES is concerned, you can see there are variety of stents, be it metallic, be it biodegradable polymer coated or bioresorbable, which are not having any metal itself. Initially, it was thought that bioresorbable ones would flood the market, but that didn't happen. In fact, Abbott had to withdraw its stent. Why it didn't happen? Because you can see the strut thickness here is much more as compared to metallic ones. Metallics can go to as low as 60 micrometer, and 60 micrometer is what we are offering. That means the Surge Anand has the thinnest strut possible, and our endeavor is now to go to 45 micron, while having all the mechanical strength and performing functionally also in the same way. Also, something to be noted is that you can have coating which is circumferential. You can see here fully coated, or it could be partially coated, only the abdominal side coated. So people have also worked in this way so that they can decrease the drug load. See, drug is required initially to take care of inflammation which is happening, to take care of the cell growth, but drug loading should not be so much that doesn't allow the artery to heal. So to balance that, you need to look at the amount to be coated. This is another stent which is Biomime by another company in India, Gujarat-based company, Merrill Life Sciences. And in fact, they have also indicated that binary restenosis was found only in 5.7% patients. In-stent restenosis, meaning within the stent, the blockage was 4.5%. But otherwise, it was little higher. And what they have is an intelligent design where they have open cells and closed cells. You can see here, open cells and closed cells. So there's again, I would say, a scope in working on the design. And our company has mastered a design where they can, after placement, the stent gets locked, so position will not change. Something like you have hard gelatin capsules. In case you are aware, there are self-locking hard gelatin capsules now. So earlier it was body and cap simple. They had to be put together. Now you have intelligent design so that they get locked in place. Nothing leaks out. Same way, stents also can be locked in place. So there are various problems with percutaneous coronary interventions. Problems like in-stent restenosis, as I've said, you've implanted a stent, but within that, maybe over a period of time, again, if the drug amount is too high, it doesn't allow the cells to grow. Net result is there is in-stent restenosis or target lesion revascularization. Drug amount is so low, that these cells go on proliferating or leading to, again, blockage. Stent thrombosis. You put a stent, but because you have a bioerodible polymer or some other metal leaching into these particular vessels, that thrombi formation takes place. And next problem is late lumen loss. You have opened up the vessel, but after a certain time, the formation, meaning your drug has eluted out, again cells start to grow and your vessel diameter again goes down. So all these problems we realized are for a variety of reasons. It could be the stent as a platform which is incorrect. It could be the drug dose which is incorrect and not balanced. It could be the release rate from the polymer which is incorrect. And it could be the polymer itself leading to these problems. So having understood what are different problems, we started working on all of these issues. And the answer, of course, was in nanotechnology. It has overcome all the current drawbacks, I must say. It has reduced the dose. We've been able to release the particles in a sustained release manner. We have seen that drug, whatever is there, is not affecting the other parts. It's releasing only in site-specific manner. At the same time, there's no local overdose. So what have we done? Obviously, onto the stent surface, these nanoparticles are coated. So once they are released by the balloon, you are expanding the balloon, the stent releases the drug particles. Now these drug particles would move on to the vessel side. Vessels, if you know heart, any vessel will have layers. So you do have endothelium, tunica media, and tunica adventitia. So we want that the particles which are there, they move towards the vessel side, they get lodged there, and from there, slowly over a period of time, they go on releasing the drug moiety. So with this hypothesis, we were working. This is just another system which got approved in 2019 and claims that it has ultra thin strut cobalt chromium, but with sirolimus. Now this one has polymer PLLA, polyl-lactide acid. So it delivers the drug over 12 to 14 weeks, and in 1 to 2 years it degrades. So this particular innovation definitely has given some important information to the world that if you want to treat peripheral arteries, this might have a solution. So there are variety of advances which have happened. As I told you, nanomaterials are being used. These materials have to be hemocompatible. They should lead to re-endothelization also. You need to increase drug delivery efficiency. You need to reduce the side effects. But at the same time, if you look at the topology, these nanoparticles should coat the stent surface very, very uniformly. So they should be of adhering type. They should lower the thrombogenicity. The same time, the vascular growth which happens, the normal cellular growth should not get hampered. So recent trends are that you are having different kinds of stents. You can see in 2017, people are using dissolvable stents, and these are the computer tomography images wherein you can see the lumen open. These were the first generation stents. But the next generation, the current generation have opened up the lumen in a much, much better way. But still, this is a long way to go. As I told you, these all are bioresorbable stents. But strut thickness is something which is still hampering their use. So we need to keep this in mind. Keep this figure in mind. We have achieved this with the metallic stents also. So these are the requirements for biodegradable stents. And I'm sure I need not elaborate on any of these because slowly, steadily, I've actually explained everything what is needed. Coming to case study, this is how, meaning on my fingertip, I've taken the stent looks when it is in unexpanded form and when it is in expanded form. People are working on nanohybrid hydrogels also now. A lot of work is being done. You can see in formulation area. Even if you can have cobalt chromium as a base or stainless steel as a base, thin strut, what is required is can you get the release in a certain way as it is required. So not only people are working on small molecules, they are also working in coating VEGF, coating angiopoietin, maybe even the genes. For certain diseases, gene delivery is a must. So if you can take it to the site and release the gene, it will do the purpose. People have not even stopped looking at the brain. Glioblastoma is a major problem. You cannot do open brain surgery every time to pump in drug. So can something be done where drug is released over a period of years? And here is the answer. Okay, so our group, we are working on many areas, but focusing on definitely medical devices and also focusing on lipid nanocarrier based drug delivery systems. Lot of technology platforms, already 20 plus technologies given to the industry. And you can see in the device category, we have actually given now, this is 9 technologies have gone in already in market. And also we have given one intrauterine device to the WHO market via Femicare. Other nano-based dosage forms also in the market. So we realized very early on that there is unmet need. Therefore, started working on it by utilizing the expertise and skills which we had in pharma. And now I'll play a video which will actually clear, and I'm presenting first stent Infinium as it is known. It's a paclitaxel coated stent. This was the first one to enter into market from Sarjanan. So definitely what you can see here is now the blood vessels and vessels which have the very, very tortuous path. You can see that this is how stent would go from the thigh to the side. So obviously it goes on the balloon, and therefore balloon which is there has to be really very thin. It has to be expanded very well without rupturing. I talked about radiopaque markers, platinum. So they are the ones which will be guiding actually when on CCTV, the interventional cardiologist is monitoring. So obviously it has to be mounted on a very flexible, short, soft tip. There should not be any injury happening to the blood vessels. Balloon, when it is mounted, it is in a folded shape because it has to expand later. Stent, when it is put onto balloon, should have low crimping profile. That means stent should be able to get released when we want. Balloon should be able to deflate and come out when we want. Obviously, maximum flexibility. So our polymer coating composition is something which we are very proud of. It's a very, very flexible one. Both ends, there are radio opaque marker bands. Now you can see it has entered the place where yellow or orange indicates a plaque, and you are actually putting in air, inflating it so that the balloon expands, and the balloon expands, stent also will expand. But when stent expands, one needs to see that all the struts be it radial or longitudinal, expand. Rotation of the junction should happen upon expansion, and that is what our company has built in, as I was telling you, lock-in mechanism. Once it is placed, it should get locked in place. There should be zero foreshortening. That means it should be in the area where it is desired. The stent should not move above the area of plaque too much. Otherwise, it will not give the drug in a site-specific manner. And these drugs are cytotoxic, so can create issues. So you need to be very, very careful. Also, after placing, there should not be radial recoil. If it recoils, there would be artery, which would again get lower millimeters in diameter. And now you can see the balloon has got deflated and taken out. What remains is stent in place, stent which is compressing the plaque. Obviously, as far as possible, there should be lower metal to artery ratio. And that's why I said thinner struts are preferred. 60 is what we have, 45 is what we are working towards. But if you can make this design with thinner struts, nothing like it. So that was about the bare metallic stents. And then after designing it, I'll tell you, it was only a paper design which was given to them. And from paper design, they actually designed the full machine, prototype machine. And then we started experimenting. At that time, even in my lab, I didn't have a HPLC. We used to outsource all the analysis, but we used to work daily basis, calls on daily basis from Surat to Mumbai. And really, we went in very fast. Imagine 1997 starting the concept, and 2005 after clinical trials putting this particular device in market. It was a Herculean task, but that's what we are, ready to take challenges anytime. So from this, Matrix was a known for bare metallic stent. Infinium was a name of a paclitaxel stent. So Infinium obviously had Matrix as a base metal, paclitaxel as a drug, and polymer base. Paclitaxel, all of you are aware, it has anti-inflammatory property. It is an anti-cancer drug, and of course is extracted from Taxus brevifolia. To the people who are in this forum, I need not tell about paclitaxel. But why was this drug selected? Because already stents which were marketed had paclitaxel. So we looked at what advantages it has. So it prevented the smooth muscle cell migration, which was a main action. Plus, in addition, gave anti-inflammatory effect. Now we all know that it is lipophilic drug. And therefore, being lipophilic has another advantage. It remains in the tissue for longer period of time. So effect is much better. And this is where our information came handy. We use biodegradable polymers for the very, very first time world over. Initial design had 4 layers, but now from these 4 layers, we have improvised the design over a period of time. And now we have 2-layer design. And the drug polymer coating, which was done, was earlier microspheres, now nano. And that's done using acoustic activation and carrier stream technology mixture. So what you need to see is the surface is uniformly coated and gives the release profile, which is biphasic. So initial one to take care of the condition, and then over a period of time, it should maintain the constant rate. So we moved that way from bare metallic stents to drug loaded stents. And this case, it was paclitaxel. After paclitaxel, of course, we moved to everolimus. We did a lot of work even for companies like Abbott. We worked on sirolimus, and now we are working in various other drug categories. There are still challenges like this. If you have to work for diabetic foot ulcer, you need very, very thin strut. You can see here the nitinol stent. So people need to look at such areas where there is a lot of focus. So as I said, we have now started working on nano, but before that it was microspheres, for which you need to do experiments in pigs. But before you do it in pigs, you need to do certain cell proliferation inhibition studies. So you can see that this is with what is there in the red is with uncoated, and with coated, definitely you could see the proliferation intensity reduced. Blood compatibility needs to be checked, biocompatibility needs to be checked. So there should be minimum leukocyte adhesion, there should be minimum platelet adhesion. So these are the studies which are done in Yorkshire swine, as I said, in pigs, and results indicated that late loss was hardly anything. And I told you late loss is an important phenomena, because you don't want again the artery to be blocked. So histological studies, I told you, if you use again too much of drug, what would happen is you would get delayed healing. If you do not use drug, the neointimal thickness, that means the cell proliferates so much that this thickness increases. So you need to have something which is drug along with polymer. So these are the results with Infinium, and these are the drugs with supralimers, which contain sirolimus as a moiety. You can see the arterial lumen opened right from beginning till end. So after having the preclinical, we went on to clinical. It happened in 11 centers in Brazil initially, and now being done all over. Even in US right now, the work is going on. The 65 plus countries which I mentioned does not include US. And once the studies are complete, it will open up the entry for US market. And then I think this company will be number one company all over world. Right now we are at number 3. So the pain trial study design included 274 patients. Randomization was done. Some had only bare metallic, some had paclitaxel coated, some had sirolimus coated, and clinical follow-up was done for 36 months. So you can see that as compared to the bare metallic stent, which is Millennium Matrix, these stents are performed better. Meaning in all respects, it is better. And that is how it is plotted here. You can see what happens, the bare metallic versus what happens with drug eluting stents. Target vessel revascularization, we don't want revascularization to happen. The incidence of that also is lower with drug eluting stent. And this slide depicts clinical evaluation up to 36 months. And you can see that 2.1 in case of sirolimus coated. That means this is the best as compared to paclitaxel or bare metallic. Texco study, I had told you, keep in mind, OCT, which was done, also indicated that our stents are performing as good as Abbott stent. And in fact, this gave many, many awards to the company, and company got known world over because ideally speaking, if you see now data, we are better than Abbott. And these are the OCT results. You can see the opening of arteries is very, very good with Supraflex screws. That's the latest stent platform which we have. Very thin strut, a different design, sirolimus coated, and this has been compared with Abbott stent. So in all the parameters, I think we should not go into integrities of this. All the parameters, when you see, it's comparable clinical data. And in fact, this was 12-month clinical outcome. And then even after 36-month clinical outcome, and now we'll be having data for 5 years, which I'll be able to put the next presentation. We have said that this is comparing as good as Abbott. So what we've been able to do is from bedside, we've been able to take the technology to the bedside. You'll be glad to know that more than 3 lakh stents have already been implanted. We have reduced the cost more than 5 folds. And then, of course, whatever are our learnings, from those learnings, we are working on it and still making it much better to reduce the cost while giving the quality stents So this is a reiterative process. You can't stop. Once you've learned something, you need to work on that something which irritates you, be it instant stenosis or be it anything else. So continuously go on working and do not change the goal. I always say you can change the ways. So as I said, now from microspheres, we are moving on to nano. And these are all the stents which we've already put in the market. So what are we now working on? We are working on nanoparticle coated ones. So you can see the nanoparticle uptake, and these are in smooth muscle cells. Definitely they are taken up to quite an extent. Even at half of IC50, they are taken up. That means we'll be able to now reduce the drug dose at least to 1/2. So that means the drug related problems will go. Simultaneously, on the strut side, we are working, we are making it thinner. I told you from 60, we are going to 45. So it will take care even of those issues. And you can see after coating, when I take SEM, these particles are still in nano range and all of them are less than 150 nanometers. So that's a great progress. In fact, we already published a paper in this. We've taken patents. We have many US patents with the company. Not only that, there were other challenges. How do we actually do the release study? How, before going on to pigs, can we know whether permeation would happen to the arterial vessels or not? So we have designed our own ways, wherein we take the aortic tissue, mount it into a tube, and with peristaltic pump, we start operating, we implant the stent, and then at different time intervals, we take it out and see how much is in which layer. I mean, such are the studies which need a lot of inputs. Still, there is US FDA doesn't have a fixed procedure. And every time we can't spend that much amount because all the studies are done in Netherlands, so we can't send them to be done on pigs, and pigs have to be again slaughtered also, later. So looking at holistic development, one on stent side, another on drug side, then on drug polymer mixtures, then on we are working on methodologies to evaluate also. So we did this particular method establishment. We have methods now wherein whatever will happen in 1 year can we know in 10 days? Answer is yes. So such things we have done. We have also quoted 2 drug combinations. Now, for example, in this case, we have quoted sirolimus and another drug which acts as antiplatelet. So this is bare metallic. We coat it with nanoparticles. Then we coat it with the plain polymer followed by nanoparticles of another drug. And you can see here the 2 drugs, their release profile, of course, we need to match as far as possible. But initial burst has to be there to take care of the condition. And that drug, of course, is anticoagulant. And therefore, you can see that over a period of time, it has given us prolongation of coagulation time. So I think with that, I would like to end my presentation and leave some scope for discussion if there is any. And I'm always happy reading quotes and putting quotes, because this is what Mr. Nanta Krishnan, who's Director General OPPI, had said, that you need to work together. So academia has to work together with industry as well as government. Then only we will realize healthcare innovations. And I believe in these words. So right from the beginning, if you have any particular project in mind, see who will be your takers. See, many times it is the other way around. Industry comes to you, you suggest, they do it at their end, and then they market. But it could be that you can start at your end also. In fact, on basis of this, what we had worked with the company, The drug was suggested by us, the polymer was suggested by us, the release pattern was suggested by us, evaluation strategies were suggested by us. So can we not suggest the combinations? So drug combinations we suggested now. The nanoparticle was suggested by us. Bioresorbable stents they wanted to go in for. The polymers were again suggested by us. So there's lots what we as formulation scientists can do. This is our lab. We have everything within the lab, be it we have 3 HPLCs now, we have Zeta sizer, we also have a master sizer, we have USP type 1, 2, 3, 4, all apparatuses. We have a ultracentrifuge. We have a cell culture lab, which you can see at the back. So all this has been created through government and industry projects, let me tell you. When I started my work 30 years back, I started working on beaker technology, got some results and started putting projects left and right to government, and happy at 80% of them were granted. But I did not lose my focus because all in our lab, we work with this motto. In fact, all the students who enter the lab are told to say this, really loudly so that gets imbibed in their mind that they need to take idea. It could be one idea which either they come up with. If they do not come up with, I give them. But that idea is what they have to make their life. They need to think of it, dream of it, live on that idea. And really, our students who are trained by different, I would say, states across India, when they come to us, they do an excellent job. They work in a very focused way, and therefore, we achieve the success what we achieve. You can see our smiling gang. This is on a Dashera day. And yes, finally, I would like to end by saying that intelligence is ability to adapt to change. So do not do the same old things again and again and again. Every one of us has been given a brain which can perform definitely in a way that there would be marvels. So work towards it, and you will see that many technologies from your end also would actually get transferred to industry. So with that, once again, I would like to thank APTI Women Forum Punjab State as well as College of Pharmacy for giving me this opportunity to share something which we have done. Thank you so much.
Thank you, ma'am. Thanks a lot for your kind words. So, ma'am has discussed about the various treatment procedures for coronary artery disease and about coronary stents. She has also talked about stent customization, various hindrances which one face in the formation or the development of stunts, and also she has also discussed with us the nanotechnology based advances in the stunt design, nano hybrid hydrogels as a stunt coating for gene delivery. Thank you, ma'am. Thanks a lot for your precious time. I just want the audience, if they are having any kind of queries, they can post in the chat box. We will unmute them. So if you are having any kind of queries from the delegates, so we will ma'am, there is not no question in the chat box. I think this whole chapter has been, lecture has been very well explained. It was so knowledgeable and informative lectures. So thank you very much, ma'am. Thanks a lot. So it's time to felicitate Dr. Vandana Patravale. So ma'am, kindly accept our token of gratitude.
Thank you. Thank you so much.
Thanks a lot, ma'am. Thank you. So moving towards our next second technical session. So we are privileged to have Dr. Manjinder Singh Gill with us. I welcome you, sir. So Dr. Manjinder Singh Gill is a assistant professor in process chemistry in Department of Pharmaceutical Technology at NIPER Mohali. His major research areas include synthesis of therapeutically important compounds with focus on anti-diabetes, anti-HIV, antimicrobacterial, and anti-cancer agents. Another area includes pharmaceutical process research and development on lab scale of drug molecules, APIs, and herbal compounds. Applications of green chemistry to pharmaceutical processes with focus on reactions in aqueous organic media, total synthesis of therapeutically important compounds and natural products, synthesis of drug conjugates and complexes to enhance therapeutic profile. He has handled various industrial research projects like 20 plus projects on scale up validation development batches of APIs such as cefoxopran, candesartan, pioglitazone, etc. Four projects on validation studies of herbal extracts of curcumin and jamun, isolation, 5 projects on synthesis of process-related impurities of drug molecules, and many more. He has guided more than 60 plus AmTech students and 7 plus PhD scholars. He has 9 granted patents, 4 filed patents, and more than 20 publications to his credit. He has delivered various national and international talks in various conferences, workshops, and seminars. So now I would like to invite Dr. Manjinder Singh Gill for his talk. Sir is going to speak on diphenyl carbonate mediated synthesis of sulfonyl ureas and amides. Please, sir.
Thank you, ma'am. Can I share my screen now?
Yes, sir.
How do I do that? Should I open my PPT first and then share it?
Yes, sir. So
Okay.
you should first open your file.
Because we mostly use Google Meet. So this is now there's so many programs.
So on screen, you will find one green folder that is share screen. After sir, opening of your file, you can go for share screen, then you can share your PPT, sir.
Can you see the screen or not?
No, sir, it's not visible. So, on screen, there is one green folder, share screen. So on your main screen, there is one green folder, share screen. So you can click on that. Then you can share your window, and after that you can share your self presentation.
To see my
So green सा वो है छोटा सा folder बना है with arrow.
Log in again, log out, and then come back.
Okay, sir, you can.
Can I continue? So by the time, can I start now? Can you hear me?
Yes, sir. Yes, sir.
Okay. So by the time the process goes for chemical manufacturing, this is an optimal synthetic process. I'm sure most of you are working with pharmaceutical chemistry, so you will realize that the process is changed with time. So the boxes in the red boxes, that's what we do. We do process R&D in the lab and then develop the process, and that information is fed to the Kiro lab or the pilot plant, and then after sorting out the processing issues, it goes for chemical manufacturing. So this is the technology. This is the most valuable in terms of API and intermediates. In Kilo Labs, as I said, the cost is not a concern, but the poor yields are addressed, and deadline is because it has to go for toxicology and PK. Pilot plants where you have to do the processing problems to be solved. Yield optimization is done, even quality is addressed. And then the chemical manufacturing is where even a small amount, a small change in any product or yield will have a major financial impact. So basically, the process is simplified from lab. This is what we do, optimization of lab scale synthetic route in the lab. And then it is simplified right up to the chemical manufacturing process. So basically the process R&D strives for improved reaction efficiency, improved yield and quality of the product, improved productivity on scale. When you're doing any reaction over 50 liter volume, that's called on scale process. Anything below that is lab scale process. So that distinguishes medicinal chemistry and process chemistry as well. The other objectives are to reduce the cost of the product and to reduce the waste generated during the manufacturing. Only few slides just to give you a flavor that how API or process developments are being done, because API production cost has two components: material cost, material of the reagents, key starting materials, and then conversion cost. That is the process how that starting materials are converted into the final product. So at times you may not be able to control the material cost, but you can change the conversion aspect and vice versa. So making changes in both, you can reduce the cost of the product and reach all the goals listed under process optimization. See, innovation is everywhere in daily life. I usually give an example that little modification of a process can make it more efficient. I think most of you have seen it. If somebody is making a huge pot of tea and is adding tea powder to that, then on the right side there's a pouch that can be filled with the tea and thrown inside, as we do when we make tea in a langar. So that makes the process efficient, because left side manufacturing, you will have to filter the tea, and at least you will lose 10 to 15% of the tea during filtration. The right hand side, you will just remove the pouch out and you don't need to filter it. So there's a cost reduction in terms of production. And this is also manifested in a tea bag that we recently used. So this type of innovation is part of the process development, which seems rather simple, but it can have very serious financial and manufacturing consequences. In process efficiency wise, we all in the lab use percentage yield. You all know actual yield divided by theoretical yield and 100. So that is also an efficiency factor when you are doing a process. But another aspect that you may learn today or may know earlier also is atom economy. So process chemists look at the atom economy, which is calculated taking molecular weight of the product divided by molecular weights of all the raw material used to manufacturing that product in 200. So this is a key factor which indicates that the atoms from the raw material has gone into the final product. So high atom economy is preferred. For example, Diels-Alder reactions, rearrangements, and catalytic hydrogenations. I hope you all understand the chemistry aspect. They got high atom economy. Low atom economy is when we have protecting groups, chiral or auxiliaries otherwise, because they are put on the molecules and they are removed after their functionality, their work is done. So that way the atom economy will be low. So that is one factor that process efficiency wise is considered by the process chemist. Then third factor is volume time output. This is when the product is in the plant. So you take nominal volumes of all reactors, multiply by time taken by per batch when A to final product, divided by whatever the output of the batch or step was in kilogram. So if the value is less than 1, then the process is accepted for scalability and its cost effectiveness. Similarly, there are environmental factors and process mass intensity that looks at the waste. I'm not going to go into the details, but I just want you to know that the approach is slightly different, that you're looking at these factors and only that technology will go further for manufacturing. Another approach that process chemists usually take when they are designing a synthetic group, this is a linear synthesis when A go to B and then C to product. Then overall yield is calculated this way, which is 51%. These are classical examples. And if you're doing convergent synthesis, you're making one xanthan, then other xanthan, and then to the product, then the overall yield will be lowest from this end and the second step. So it will be higher. In complex cases, if you don't even get a high yield, as is expected by convergent synthesis, this methodology allows you to work on xanthons at the same time. So definitely it will be a fast process and there will be less loss in terms of if there is any failure along the way. For example, if you lose C, you will have to go back to A. In this case, if you lose even one, then you go back to the other one. Therefore, the convergent approach works very well for complex molecules. And nowadays, the drug molecules are becoming even much complex these days. So in the synthesis, usually if you have a low yielding steps or non-selective reactions, you put it in early in the synthesis. That is a common approach when you are designing a synthetic group. Or if there is any expensive component in terms of reagent or activity, then you put that part later in the synthesis. Okay. So an improved synthetic process, which is a key for any process, will have high yield, high atom economy, convergent synthesis. Some terms that you may remember, that's why I'm not going into details, but just saying telescopic workup, that you don't do the work and you don't do the workup and move on to next step. Fewer number of steps, of course, you avoid protecting groups because low atom economy, you avoid adjusting oxidation states. Sometimes we have to do chemistry, not on alcohol, then a ketone and then back to alcohol, something like that. Incorporate NTO and stereospecific reactions. Use rearrangements, again, IA, use double reactions. Catalytic transformations are required. You need to avoid hazardous and toxic reagent. If reagents are hazardous and toxic, so in a plant, you need more time to handle them and more time in a manufacturing plant, higher the cost of the product. So just by changing non-hazardous, just putting non-hazardous reagents or solvents will make a process an improved process. Use neat reactions or green solvents. utilize solvents to isolate products. Sometimes you can utilize the solvent to isolate the product. On scale, when you're doing in the plant, liquid reagents are preferred over solid. In the lab, it's other way around because liquids can be pumped in to the reactor and they can be measured accurately. Plateau type of reaction conditions are preferred. Plateau type of reaction conditions are when there is no change in the product outcome in terms of production and quality. If there is a delay in, if there is condition varies from one range to another. Point type of reaction conditions are when there is a slight change in any reaction conditions, the quality and the yield will be changed. So they are not preferred. So you need a rugged forgiving process to be developed for scale up and manufacturing. Basically, green chemistry principles, those are listed for green synthesis, are all applied to for process development. But the major difference is, as you can see on the right hand side, this is a lab setup and this is a reactor. In the lab, you can remove the flask and do filtration the way you want to from one place to another. But the reactors, this is a vertical cross section, is a stationary vessel. So you cannot remove the reactor. So that's why some of the things are different. So there is an agitator. You can see the cursor. There's an agitator on the top and impeller at the bottom. So it stirs. There's a jacket that will heat and cool. This is a manhole, not too big to add input of raw materials. Then there's a condenser that will go and you can remove the product at the bottom. So it's a fixed stationary vessel. So you have to develop chemistry in the lab, keeping this in mind. Although process R&D is done in the lab first, and then you do. There are baffles, the rods sticking on the both side, because the solvent may form a vortex, and you have to avoid that. And there's a limitation to temperature range. For manufacturing range, ideally minus 40 degrees Celsius to plus 120 degrees Celsius. Any temperature higher and lower will increase the cost of the production. No column chromatography that we usually do in the lab. Even no recrystallization. What is done, you make a slurry in a solvent and then filter it off. If that improves the purity of the product, or crystallization. No recrystallization. Charcoal treatment, which we commonly do in the lab, is avoided because you will lose the product and it's an extra step, extra time, extra cost. Azeotropic distillation is preferred because that way you can dry the solvent, you can remove one solvent mainly and then switch the solvents. So these are typical operations, only right up to here I will say that those are used in the process chemistry. So as mentioned earlier, we are working in anti-diabetic area and sulfonylureas are oldest drugs, but still they are better in terms of safety, efficacy, and cost-wise. They are dual in actions, scriptobacillus and sensitizers. So even recently, there has been other applications in terms of their therapeutic activity. And there are some of the sulfonylureas, those are even working against hypertension, cardiovascular diseases. So that was the interest to tackle the sulfonylureas one way or another. So this is a list of first generation sulfonylureas. This is the sulfonylurea functionality you will see. But on the urea side, there's alkyl chains mostly alkyl chains. And now the second generation was a bit more complex. But even then, on the urea side, it's in alkyl chain all the time. Now the benchlamide and glycoside, they are commonly available in the market. Then there's a third generation, which is glimepiride. You can buy in the market also, but it has two different functionalities: sulfonylurea as well as unsymmetrical urea. So if we look at the processes, the way they are synthesized, they are actually synthesized using isocyanates, either sulfonyl isocyanates or otherwise isocyanate reactions with the sulfonamides. And then isocyanates, in both cases, they come from amines and phosgene, a gas, toxic gas. Isocyanates also reminds us of Bhopal gas tragedy. So handling of these chemicals on scale is hazardous and difficult, and hence a costly affair. So if it can be replaced with something benign, then the process will be considerably improved. For example, these are the issues with the known processes: hazardous, irritant, moist, sensitive, and also expensive reagents to use. So alternatively, if something like a carbamate can be used with an alkyl chain, that will make the process safe. And for that, we picked diphenyl carbonate, which is a safe carbonate, solid in appearance, and is not very costly. There are a number of other carbonyl sources, but this was picked mainly because looking at the cost part, safety part, handling part, availability part, as that the part of the process issue is. Because these are the sulfonyl ureas because carbamate is a surrogate to an isocyanate. At higher temperature, it will decompose to generate isocyanate in situ. So if it doesn't work one way or another, it can be regenerated in situ and then utilized the way it has been done in the past. But when we did our initial experiments using the DPC with the amines, it gave mainly the urea, and there was no sign of carbamate formation. The reason being that amine being more basic than a phenol, it will readily replace the other phenol as well to give the urea as a major product. So fortunately, chemistry is try, so you continue to try, and at times even random steps will be giving you the desired products. So when this reaction using N-butylamine was carried out in hope to get a carbamate or a urea as a byproduct, the left side table you will see when THF, acetonitrile, dichloromethane acetone was used. The main product was urea. There's a small amount of carbamate formation. But when the reaction was conducted in water only after 8 hours, 80% of the carbamate was formed. Mainly, the success of this reaction relies in doing reaction near insolubility. So the carbamate actually crystallizes out, and that stops further reaction of it with the amine molecule. So once this is the first time when this type of carbamate was synthesized. So we filed a patent about 5 years ago. Then reaction conditions were optimized, taking THF water, and although the yield is not very much different, but reaction time has gone to 5 hours with the 10% of THF in water mixture. DPC quantity was also optimized with 10% excess of DPC. Reasonably good yield was obtained to get N-alkyl O-phenyl carbamates. They're key intermediates in many other chemical synthesis, but we looked at a number of primary amines and the yield was reasonably good. This patent was the first one we filed and it has been granted last year. So because this was the first time this type of carbamates were synthesized. Utilizing this chemistry, tolubutamide, first generation sulfonylureas were synthesized readily by taking a sulfonamide and a carbamate and triethylamine after reflux in a good yield. Likewise, another first generation carbamate was sulfonylurea was synthesized this way. Now, this carbamate synthesis was explored in many ways. We were able to make sulfonylureas. We were also able to make mono-substituted ureas. They're not easy to synthesize otherwise. We were also able to make unsymmetrical ureas. These intermediates are all part of many drug molecules and semi-carboxyl. So we targeted large number of drug molecules and filed a number of patents. 3 of those related to this work got granted last year and the year before. But then earlier on, when we filed our first patent, we tackled a 3rd generation molecule, which is a glimepiride, and it is being used in large number of countries. And synthetically, it was challenging because it has 2 unsymmetrical urea moiety as well as sulfonylurea side chain to that. So this was synthesized and also published in Synlet at that time. Taking a lactam 5-membered that can be activated with the DPC to get to this carbamate, although yield in this case was on low side. As I mentioned, any process that has low yield early stage wise can be acceptable, and then reaction of that with the amine intermediate gave unsymmetrical urea part, and then sulfonamide side of that was reacted with another carbamate that was again prepared from this cyclohexylamine with the use of DPC in THF water. These are typical reaction conditions. These are slightly different. And then both were combined to give glimepiride in 38% overall yield. We're still working on this to improve this, and this is ready to go to the next level, that is to the kilo lab for further exploration, because this is one of the main drug still available on the market. Similarly, 2nd generation, so many of those were made, but then glibenclamide is another drug molecule that is on the market by taking this benzoic acid derivative. It was activated with the thionyl chloride typical way, and then an amide bond was formed. Then taking another cyclohexylamine, making a carbamate using typical our methodology. Then both combined again. This was also more than 40% overall yield. And this methodology or this process was patented last year. It was a provisional application. So by the time we have more to add to it, it will be filed for final bid. So after synthesizing most of the sulfonyl ureas, one extension that came to our mind, that can it be an activated to form an amide bond. For example, when we take a carboxylic acid and react with amine in the presence of DPC, will it give an amide? Because amide is also present in so many drug molecules. Although the byproduct could be that it's a carbamate, because DPC will react directly with the amine. And if that works, then we will have an improved process for amide coupling as well. So that will open up an other avenue for us to explore. DPC is, there are many advantages of that. As I said, it's stable, it's safe, it's alternative to toxic and costly coupling reagents if it works, inexpensive, easy to use, sustainable, and so forth. So amide bond, as you know, there are so many drugs and drug intermediates. Those got the amide bonds, as I have shown this in blue color. Actually, these all are already have been synthesized in the lab. So that's why it's cut and paste from that side. And they say 66 of 200 best-selling drugs in 19 had at least one amide bond. And in some cases, there are more than one. So usually the amides are synthesized by three different ways. You can couple an amine with the carboxylic acid using a coupling reagent, for example, either activating with thionyl chloride, oxalyl chloride, CDI, DCC. All of us have used this in the lab. But some of the reagents are very expensive and some of the reagents are even toxic. There are catalytic condensations where metals are used, boron-based catalysts are, they can combine a carboxylic acid with an amine. And likewise, there is thermal amidation where heating at high temperature or in a microwave reactor, you can get them coupled with the elimination of water to give an amide. But there are limitations to these reported methods as well, because coupling reagents are expensive. Although thionyl chloride, oxalyl chloride, POCl3, they are not, but they are corrosive and hazardous when you handle it. The pilot plant I had taken care of for 10 years, most of the piping of the plant got corroded only because of these two reagents. So they generate HCl and they're really corrosive in nature. Other coupling reagents, as we have already used, DCC, EDC, DIC, they are expensive and toxic. And the byproduct in these cases is mostly urea, and which is not easy to isolate from the desired product. And it produces large quantities of waste. So as from a process point of view, even the E factors are not in favor, or even the atom economy is not in favor, because large part of, for example, DCC will give you urea, so most part of it is wasted in the process. Thermal amidation, those are harsh reaction conditions, so not cost effective for manufacturing. As I said, any higher temperature over 120 degrees Celsius will require special heating and it will cost. And this also forms large amount of byproducts. Thermal amidations never go to higher yield. There is always some of the products, reagents left unreacted. And catalytic amidations, difficult purification because there's a metal waste, and that makes it overall a costly process. So overall, all these known processes have low atom economy. So that's why there's a need if you can simplify the process. And there are so many drug molecules that got amide bond in there. Very recently, there is a huge review article in OPRD, Organic Process Research and Development, 130 pages or more, that has listed the coupling reagent for amide bond formation on large scale, or on scale as we say. You will see that some of the expensive reagents are used because they give the clean product. For example, thionyl chloride is not expensive, but it must be used in the early stage of production, and these two in the later stage of production. So some of them are expensive, some of them are corrosive and difficult to handle. Even then, having that many reagents, every reagent has its positive and negative aspects. You can have another reagent in there if it's reasonably improved methodology, gives it improved the process for the amide bond formation. So interesting part was that when we took this benzoic acid and treated it with benzylamine in the presence of a base solvent DPC and heated it to 60-70 degree, hoping that it may give an amide. The interesting part was actually discovered by chance, you can say. If all of it's added one pot, as you can see on the right hand side column, carbamide was the main product, 85 to 70%. If everything was added together and then the product was isolated. But if it was added sequentially, for example, you took the acid, you added DPC and a catalytic base and let it stir, followed by addition of the desired base, the desired amine that we want to be forming a amide bond. Then you see the amide formation there. So for example, some of the organic bases, they gave 10 to 40% of the product. But some of the organic entry 5, 6, and 7, there was no detection of any product. Even lithium hydroxide, nothing happened after 24 hours. Pyridine reflux did not give any of the product. But when triethylamine gave almost no product, DMAP gave 80% of the amide that was expected. So DBN also gave, but the yield was low. Imidazole had reasonable yield, but the carbamide intermediate, that 10 to 15% was also isolated. So in this way, the entry 11 became our inspiration or the main methodology to explore it further. So when optimization of base quantity was done, with 1 equivalent, the yield was same, 0.5 equivalent, the yield was same. Lowering of any molar quantity of that, the yield dropped. So entry 2 was taken as optimized base concentration. Similarly, with the imidazole, there was not much difference in entry number 1 or 2. So that was also just done to see if amine has a role to play. So in terms of cost, in that review article, as I mentioned, there's a review article very recently published. You will see the cost of DCC. We're taking Sigma Aldrich prices here for 1 kg, although the bulk prices are way low. For example, for 20,600, you may be able to get 15 kg of DCC when you are going for the bulk price. But simple comparison will show that DPC is of very low cost. So cost-wise, it falls on the low side of, this is a per mole cost. And even if we include the cost of DMAP, which is 50% mole equivalent used, then even it's somewhere right there. So cost-effective reagent, as it turns out to be, can be utilized for the synthesis of other API and other intermediates. So making use of this optimized process, DPC, DMAP, THF 60 to 80 degrees Celsius, 3 to 8 hours, sequential addition, many primary amines gave a reasonable yield of the product. As you can see, some of them are in high 80s, but some of those are in 70 as well, because methylamine also evaporates from the reaction pot. So this low side can be attributed to that. But these are isolated yields, no purification by column chromatography. These are crystallized material coming out from the reaction flask, the way process chemistry will approach these syntheses. Likewise, secondary amines were utilized, and they also gave a reasonable yield of secondary amides. Then aliphatic acids were taken, and then they were exposed to aryl or aryl alkyl amines, and then the yields are acceptable because this is a one pot reaction, no activation as such, and the product is isolated. The other byproduct is the protonated amine that can be washed off, phenol and carbon dioxide. So the purification is relatively easy to perform, and purification is a bigger component when it goes to manufacturing plant or on scale. Similarly, some chemoselectivity was observed when we had a diacid, got only mono in 66% yield. This may have gone by anhydride cyclic intermediate. We don't know yet. This is very recently done, and a patent has already been filed on this methodology. Similarly, different amines are used there to get monoamide formation, while other is not touched. So it devoids any protection, deprotection. So ideal for scalability or on large scale. Similarly, N-protected amino acids were also subjected to this amidation process, and amine was protected, then we got the amide. Likewise, this was protected and we got the amide. Some of the amides are rather complex because they are part of some of the intermediates. Those are being used in some of the drug molecules. So some utilizing this methodology, drug molecules were synthesized. Acetaminophen in overall 69% yield. This may not be a process for scale up, but this is another alternative using the para-hydroxyaniline and acetic acid. Usually what they do is they take para-hydroxyaniline and use acetic anhydride, which is a restricted material in any country nowadays. Similarly, Varinostat is another anti-cancer product. And because of the selective amide formation, we were able to do another selectivity on the other side to get this in one pot, two-step process. Patent on this is pending at the moment. Likewise, another product, which is a basically antidepressant product that can also be prepared from reaction with an amine and the corresponding parachlorobenzoic acid. So in terms of a comparison, usually some of the reagents are acetic anhydride, phenyl chloride, or expensive coupling reagents, fluoroformates, or even metals. This is very simple, cost effective, and environmentally benign. No column chromatography, hence making it scalable and further use in synthesis of some other drug molecules, which we have done. This is another anti-cancer drug. And the key intermediate was prepared using this methodology. Now this is being transferred further, and as you will see, there is also a urea moiety in the product, so that will also be made using DPC as an activator or as a carbamate. Similarly, another sulfonylurea, which is a glipizide, that intermediate was prepared in high yield. There is no manufacturing of glipizide in India at the moment, so this is a commercial product. And that will go likewise. We did having another carbamate from one end and this sulfonamide. So in a single step, it will give the glipizide. This is underway or may have already been synthesized. Similarly, galvanclamide, as we saw earlier, we used an acid, then activated it as an acid chloride. Then amine was introduced to give an amide, but this process gave in one step and rather high yield of the product. So the galvanclamide final full patent will have this information in there too, because this is further improvement of the process to go to the desired sulfonylurea. Actually, the real utility of this methodology came when we were making glipizide, which is shown on the right hand side. There's an amide bond and there's this sulfonylurea moiety in it. When we had this acid and we tried to couple it with the amine, and this is our own methodology, but using different known reagent. At that time, we did not know that this can work. So thionyl chloride, there was no detection of the product. EDCI, which is a coupling reagent and expensive, we were able to isolate 20% of the intermediate. CDI, commonly used, 40%. DCC, 56%. So because there is now the ring is a heterocyclic, so it may have a role to play, but after developing the methodology, when we do DPC, DMAP, our typical sequential addition, 83% of the intermediate was isolated. So this actually emphasizes the importance of this methodology, that if others are not giving you the right productivity, you can utilize it, but operationally it is a rather simple process to go for. So in terms of possible reaction mechanism, we can propose the following. When we do one pot addition, as I said, you have a DPC, you have amine, it goes by an intermediate that is a carbonic anhydride, which is actually, this portion is from DPC and this is acid. But in the presence of amine, it reacts with this intermediate to give a carbamate, which we know earlier doing it, and we got mainly when we did the one pot addition. But if it was added sequentially, as I said, you take benzoic acid, add DPC, DMAP, or imidazole, solvent 60 degrees Celsius, let this form. Once it is formed, it will lose a carbon dioxide and will go to a phenyl ester. And that phenyl ester will react with the amine to give an amide. So this is a possible reaction mechanism in terms of explaining different outcome with different addition modes. So in process development, there are so many factors that can actually play a role in optimizing a process. And support to this proposed mechanism came when mass spectra of reaction mixtures were run. For example, in one pot addition, when MS was obtained, you see the molecular weight of the carbonic anhydride as well as the carbamate, which is in this case methoxybenzylamine. But when the sequential addition was done, then carbonic anhydride was formed, and there was also molecular weight corresponding to the phenyl ester, which eventually after addition of the amine disappeared and gave an amide product. So this supports what we proposed earlier in this. And at the moment, I asked my student to look at this aspect. Is it easy to make phenyl esters using just this methodology as compared to the known processes? Because that could be another utility of this reagent. So in summary, this is a very simple, cost-effective, environmentally friendly, if not absolutely, relatively speaking, reagent and methodologies that has been developed. Operationally, they're very straightforward and significantly environmentally benign as well. So several commercial drug intermediates and products has been synthesized with good to outstanding yields. In most cases, as I said, the byproduct is phenol and carbon dioxide. So in terms of atom economy, in one of the referees in green chemistry actually objected to that, that there is you're losing 2 phenol molecules, so your atom economy is on low side. But during the production, you can always recycle the phenol. Phenol can be a byproduct coming out of the process. So that is not a bad thing and will not add to the cost. So no column chromatography in all those syntheses, and that makes these processes suitable for scale up and further. So I took this work from 2 of my PhD students, Dinesh Kumar Tanbar. I need to acknowledge them. He completed his PhD in 2017. That was one of his projects, sulfonylureas. And Prachi Ramthake, she's a PhD scholar in the lab at the moment. And this is one of her chapter that is on making amides and their utility in manufacturing different drug molecules. And of course, Sniper Mohali for financial support. And thank you for giving me a chance to discuss this with you all. I hope it was of some use. If not,
Yes, sir. Definitely.
Thank you.
Hanji, for such a wonderful session. Sir has talked about practical applications of sulfonylureas along with their synthesis. You have very well highlighted the synthesis of carbamates with their optimization studies. Then you have explained carbamide-based synthesis of different compounds such as glimepiride as well as glipizide, as well as DPC-mediated amidations. Now the session is open for the queries. If anyone is having any kind of query, he or she can post their question in the chat box.
No, you can even ask me if you want to.
Hanji, we will unmute anyone from the audience. I think, sir, there is no query.
Okay.
Hanji.
Okay. Thank you so much.
Thank you, sir. Thank you again. Now it's time to felicitate Dr. Manjinder Singh Gill. Kindly accept our token of gratitude, sir.
Thank you so much. I'm happy to get this.
Thank you, sir.
Thank you. Bye-bye. Take care.
Thank you, sir. It's privileged to have Ms. Kavita Kanoje with us. I welcome you, ma'am.
Good afternoon, sir. I can see Tugu, sir.
Sir, please unmute yourself. Dr. Kapoor.
No, I'll call her. That's okay. You continue with this. Take care. Bye-bye.
Ma'am Kavita Kanoje is an intellectual property professional, qualified patent agent since 2013, co-founder and designated partner at Path to IP LLP with extensive experience in the prior art search and analysis, novelty and patentability, technology landscape report, FTO analysis for patents, invalidation, patent mapping, etc. She is well versed with Indian patent law and actively involved in drafting and filling of patents, trademarks, industrial designs, as well as copyrights and GI, etc. in varied field of technologies like pharmaceuticals, chemistry, engineering, biotechnology, FMCG, etc. She has basic knowledge of US patent law and examination procedures. Currently, she is working on projects and innovations for the inventors, technologists, startups, or companies to help them secure IP protection for their ideas, creations, inventions, and thereby providing support in the research and development activities and business growth. She did her M.Pharm in the year 2000 and B.Pharm in the year 1998 from the Department of Pharmaceutical Sciences, Nagpur University, Nagpur. Her professional experience include she is a IP professional, co-founder and designated partner of Path to IP LLP Pune, India since 2021 August. She was a woman scientist or knowledge provider at CSIR URDIP from July 2019 to June 2021. She holds the position of patent analyst at CSIR URDIP since Jan 2013 to June 2019. She has qualified Indian Patent Agent examination in the year 2013 and she secured 72.4%. She has also qualified various WIPO distance learning courses. She is a recipient of Government of India scholarship for the IP training. She also underwent a structural training program from the leading IP experts and acquired hand-on experience with the advanced patent database. She has worked as a lecturer at the Department of Pharmacy, Kempty, Nagpur from September 2000 to April 2004. She has also joined as a lecturer at the Department of Pharmacy, Anandpuria College, Alampur, New Delhi from September 2004 to April 2005. She was associated with Gannett Solutions, Nagpur, for 3 years from Feb 2007 to December 2010 in the medical writing for the various pharmaceutical products at different pharmaceutical companies. I welcome you, ma'am. Now I would like to invite Ma'am Kavita. Ma'am is going to speak on the topic role of intellectual property rights in the pharmaceutical industry and research. Please, ma'am.
Yeah. Hello, ma'am. Thanks for this brief introduction, and thank you for giving me opportunity to be a participant in this event and to enlighten us with the multidisciplinary avenues that are there in pharmaceutical industry. So with this, I would like to proceed with my topic, that is role of intellectual property rights in pharmaceutical industry and research. Am I audible? It's
Yes, ma'am, you are audible.
so just let me share my screen.
Yes, ma'am.
So please, could you please confirm whether my screen is
It's visible.
visible? Yeah.
Kindly put it on slide mode, ma'am.
Yes, yes, yeah. Yeah. Is it
Ma'am.
visible now?
Anji, thank you.
Stop sharing. So with this, I would like to proceed with the today's topic, that is role of intellectual property rights in pharmaceutical industry and research. So as you have already given my introduction, wherein you have introduced me as an IPR professional. So at Part 2 IP, we provide all these services to researchers, innovators, organizations for protecting the intellectual property, intellect that they are having, the innovations that they are doing in a particular field, whether it be in the field of research or in a specific pharmaceutical area domain. So with this, I'll start with the topic. So yes, so the agenda for today's session would be what is IP, types of IPR, importance of intellectual property rights, and the intellectual property rights in the field of pharmaceutical industry and research. We would also be discussing about few case studies that talk about the importance of all these IPs in specifically in case of pharma. And we will also have the session on and discussion, one-to-one discussion, if any questions are there from the participants. So I would be touching upon all the forms of IP, so that those who are already aware of these intellectual property rights and their types, they will also get refreshed, and those who are totally unaware of these aspects, they will also have a brief introduction to all these forms. So first of all, what do you mean by intellectual property? The word property, as we know in a conventional definition, is something that we own, that is having some value, some assets. So intellectual property could be a value, and it can be considered as assets also. So there are two types of properties, tangible and intangible. So we are all aware about the tangible properties, that is land, machinery. You are having a company, you are having certain instruments there in your company, then your bank balance, your hard cash money that you are possessing, you are having a four-wheeler, two-wheeler. All this comes under physical property, that is tangible property, that could be touched and felt. But intangible property is a property that we cannot see, we cannot touch, but that exists and that does have certain value. It is also called as an asset. So means it is a valuable property. So that's why they are considered as assets for any company, assets for any organization, or even assets for a particular inventor who has protected his intellectual property. So under this comes the patents, trademarks, the geographical indications, then the copyrights. All these things comes under this intangible properties that we would be discussing today. It also includes customer lists, the way of conducting any business, operating business models. So all those things will also be considered under intangible property. So with trademark also, the goodwill is there, the brand equity, brand power is there. So all types of intangible properties are the properties that we cannot touch or see. They do not have a physical existence, but they do have value if they are protected in a proper manner. So what is IP? IP, as the word suggests, intellectual property. Intellect means something that is arising from human mind. That is any idea, any innovation, any creative expression of idea. So all these things comes under IP. And these are the properties that are valuable, and we should protect them to extract the value of that property. And this gives the protection as well as help in exploitation and control of the property once it has been protected or registered under certain concern authority. And by definition, as per the World Intellectual Property Organization, the intellectual property could be defined as the exclusive right granted by a state, that is a country, to prevent others from using, manufacturing, distributing inventions, processes, applications, new or original designs, trademarks, new plant varieties, databases, artistic and literary works. So by definition, it includes all the forms of intellectual property that could be protected by a country for that person who is owning that property. The person is also called as the right owner or the right holder of that property. So, and the importance of IPR. Why IPR is important? As today we are talking about the multidisciplinary avenues in pharmaceutical industry and research. So IPR is also one of the very important form of what we can say aspect that one should consider while discussing about the multidisciplinary avenues. So IPR are the rights that are associated with IP. Whatever IP you are having, the rights would be allocated to you, assigned to you based on the type of protection you are seeking for. It acts as a strong tool to protect invention, time, money that you have invested, or the inventor or the researcher or a creator has invested in that form of IP. So that is called as ROI, that is return on investment of your time, money, efforts. And as you are getting return on investment, it will again further encourage you to invest, to create further, to innovate in more what I would say advantageous manner. So that would be beneficial for all the other people in the society. And it all, as all the forms of intellectual properties rights that are getting registered are published by the government authority of a particular country. So that's why they are also providing a huge source of information to general public about what are the various creations, innovations, designs that have been registered, that are now evolving, newly existing in any particular technology area. Except for trademark, all other forms of intellectual property are published. And with all these publications, it also provides the economic growth of the country by promoting healthy competition. As the competitor gets to know about this is the way the other company is evolving or innovating on a particular invention, then they also try to build upon something new to that. With this, there will be encouragement for further development, further research, further innovation, and it would also encourage industrial development and economic growth. So, and if large variety, huge variety of material, products, systems, designs, or creations are available in market, are available to public, then it will facilitate the domestic industry growth, and the customer, the people who are end users of this property would also have a choice to select as per their requirement the various forms of products that are available in market. So a diversified market is available, and with that there is a domestic industry growth and culture, and along with that it also attract the global customers, and thereby international trade is also promoted, and various treaties are offering such type of multilateral protection as well. And if one has protected the intellectual property, then they can have it as a tool for handling the infringement cases. Infringement means unauthorized use or unauthorized access of someone else to the property that has been protected by you. So that infringement could be stopped, that could be monitored, that could be prosecuted further, litigated further, if you are having proper protection of your intellectual property. It also includes the piracy, as you can see in the copyright protected artistic works and other things, and unauthorized use. In case of pharma, it may be the counterfeit drugs that are available in market. So all these things could be protected, and your rights could be enforced if you are protecting it in a right manner, maintaining it in a right manner, exploiting it to the commercial extent that is giving you most value, that is most return on investment. So there are various types of IPR. They could be broadly described or distinguished into two types, that is the industrial property and the literary or artistic works. So, and other forms of properties that are of specific particular technology domains include plant varieties and farmers' right, which are mainly for the agricultural area. Then geographical indications, which talks about the various aspects of the geographical implications of that particular geography on that product arising from that geography. Then trade secret and SIC, that is semiconductor integrated layout designs, which is mainly in case of the electronics domain. And this industrial property, as the name indicates, it would be applicable to the industry for a particular product of a concept or idea, which would be used for commercial purposes, that is for commercial production, commercial exploitation, commercial gain. And these industrial properties could be protected, and there are various forms of protection available, like patents, trademarks, industrial designs, copyrights, in geographical indications, trade secrets, and SIC designs. So we would be just discussing about all these types in brief. What is a patent? As we say that we might see on certain product that it is written it is a patented technology, or sometimes we observe that they write patent pending. So what does this mean? Patented product means it has been protected by a patent. It has been registered as a patent in a particular government authority, that is the patent office of that country. So what a patent does? Patent is an exclusive right that is granted by a country to the owner of the invention to make, use, sell, manufacture, and market a particular product. So the technical inventions, the inventions that are in the area of technology, science, they are protected under patents. So all the aspects of that technical technology could be protected under patents. So this form of IP is considered as the most strongest form of IP, which gives protection for the technological inventions in a very, what I would say, robust manner. And the criterias are also set for passing or confirming or complying with the patentability, that is the novelty, inventive step, and industrial applicability. So for getting a patent on a particular invention, that invention should be novel globally. So the novelty is judged on a global basis, not on a territory wise basis. Means if you are having some invention, you want to see whether it is patentable in India, still you have to see whether it has been published anywhere else in the world. If it is there, then it does not comply with the patentability criteria. It should be new globally, and for that certain tests are to be considered. So novelty search, patentability searches are done to check for the novelty of the invention that you are expecting or you are interested in protecting under patents. It should have inventive step, that is another criteria for patentability. That means it should not be obvious to any person skilled in the art. That means a particular person who is also working in the same technology domain. So if it is obvious or obvious to try concept, then it fails the patentability criteria. It should have some advancement over the technology that is already existing. So inventive step would be complied only when there is certain substantial advancement in the technology that is showing some economic value, that is giving certain growth, or you can say advantageous effect to other previously existing inventions in that area. And the third criteria is the industrial applicability. That means it should be applicable industrially for commercial exploitation. So it is also called utility, or utility of that patent, that is industrial applicability. Novelty is the newness of invention, and inventive step is the non-obviousness of a particular inventive concept. So these are, if these three criteria are met, then only a patent can be granted for a particular invention. And the patent rights are territorial in nature. That means if you want to secure your invention under patent in India, then you have to file for patent in India. If you want to secure your patent in other countries, like you want to secure your patent in US, then you have to file your patent application in US. So for the country where you want to protect your invention, in that country you have to file for patent. It can be done through international filing, that is through WIPO, World Intellectual Property Organization, or it could be done as a normal what we can say ordinary patent filing in a particular individual country. Or there are certain treaties which also take part, or certain conventions like Paris Convention is there. So this also helps in filing in particular different countries under a same convention. So these are the territorial rights which could not be transferred. If you are getting patent in India, that does not mean you have the right in other countries. For securing right in all countries, you have to file separately in that countries, or apply for patent in that countries. And so why these patents are required? As we have already discussed, that IPR gives incentives or gives encouragement to the innovators, to the creators. Similarly, patent gives incentives to the individuals for recognizing their creativity, and it also encourages innovation. And as it is providing rights to the owner, it is considered as a negative right for others, because others are stopped, that are negated from using the patented invention, manufacturing, selling, or any other form of activities that are associated with this patented product or a patented process, so that are not allowed without the consent of the patent holder. And it also is done after approval from the competent authority for public use in various cases. And the main thing that is required for getting a patent, along with the three patentability criteria, is the public disclosure. So if you want to get a patent on your invention, you have to disclose the entire invention in the patent filing application. That is a complete specification, you can say. So in that application, you have to disclose all the invention aspects in description, in claims. So if you are describing and disclosing the invention in entirety, then only you will get the patent. So you have to disclose it. And the term for patent is 20 years from the date of filing. Means if I am filing it today on 18th August 2022, so it will be there for 18th August 2042. That is 20 years from the date of filing. In case you are not continuing with the maintenance fees, then that could be reduced. As you know, for tangible properties like land and all other things, we do protect these things by applying a fencing or something like that, keeping a lock on that. Similarly, for the inventions, you can consider patent as a fencing, which will not allow the entry of others, use of your invention by others without your consent, without your permission. And that permissions could be in the form of licensing agreements or joint ventures or any other form of agreements between the two, based on the mutual consent. And for these, there are certain acts and rules that govern how this patents could be handled, how the filing procedure could be done, what are the consequences of filing, infringement, and way of restoration, or how to exercise the power of a patent. So all these things are governed by Indian Patent Act 1970 with various amendments thereafter, and Patent Rules 2003 with further subsequent amendments. And in India is also a member state to other international treaties, which include the World Intellectual Property Organization, Paris Convention, Patent Cooperation Treaty, and Budapest Treaty. So there are several countries which are signatory to these organizations, and the rules set up by these organizations, international treaties, have to be followed by the signatory countries. So with all these rules and regulations, there are separate rules and regulations that are national laws for patent, and along with that there are international laws that one has to comply. Then with this is the framework of what is a patent, and how the patent could be used, how to read a patent, what are the things that are included in a patent. It are the title of invention, as we have any journal article that we read on in different scientific journals. It has a title of invention, field of invention, background, that is to which technology it is there, and what are the different prior arts, means what are the previously published things available to the public domain. Then what are the drawbacks of that, and how that drawbacks could be overcome by the invention that you are trying to propose. So it will give the summary of invention, then the brief description of drawings, which is mainly in case of certain medical devices. It may be in case of engineering inventions like that. And with that, there would be a brief description which talks about each and every aspect of that invention in a very descriptive manner, step by step, with various embodiments that could be done for possible working of that invention. So several embodiments are given, which are considered as various aspects by which the invention could be worked out. And if needed, there are certain examples, that invention has been worked in, or experiments have been carried out, some testing or evaluation have been carried out, then those are given in the examples to support the invention, the working of invention. Next is the trademark. So a trademark is also considered as a very powerful form of IP. Trademark, as the name indicates, it is a mark which would be helpful in your trading. That is a brand name, brand creation, brand equity. So all these words are related to trademark. Means you while you are trading, if you are having certain mark on your product, then that would give an impression on the customer to identify your goods and services from others. So it is a distinctive, recognizable sign, design, or an expression that provides exclusive right to the owner of the mark to use the mark to identify goods and services. And the brand name is a form of trademark which acts as means it is a name. The trademarks could be word mark, it could be a design mark, it could be a slogan. All these things could be included in a trademark. So brand name acts as a visiting card that promotes the image of a company, any company, and the range of services and goods that they are providing. And it distinguishes and identifies the products and services of a particular source from those of the others. So if different companies are acting in a particular area, then a consumer can identify each and every product based on the mark that has been given on that. Like it may be a logo, design, a slogan, a color scheme. So all these things comprise a trademark. There are other marks as well, which are well-known marks, which means may not be registered, but they have been used from long time. So they are well-known marks, and then they can register as well. And there are certain associated marks, service marks, certification marks, collective marks. So all these forms of trademarks could be useful in purpose of trading. And as for patents, the term is 20 years. Here the term for trademark is 10 years, which could be renewable further 5 years from the expiration of that 10 years of registration. And it is considered protected under the Trademark Act 1999 in India, and internationally it is protected under the Madrid Protocol relating to Madrid Agreement. Means there are several agreements, treaties, committees that were formed for protection of each form of IP to comply with the certain rules and regulations internationally. So these type of agreements were there. And there is one treaty like Trademark Law Treaty that was formed in 1994. So as you can see on the right hand side, you will see the well-known trademarks like the swoosh mark of Nike, that is just do it, then Adidas, impossible is nothing, MACD, Coca-Cola. So these are all the examples of trademarks we observe in our day-to-day life around us. And the most classic example of this could be the Coca-Cola bottle, wherein the name of Coca-Cola, that is protected under word mark. The shape of bottle is protected under the shape mark, that is the design mark. And the it also has one more IP, that is the formula of that Coca-Cola, that is protected under trade secret. Then logotype marks are the swoosh mark, MACD mark is the symbol mark, and I am loving it, a slogan, that is also protected under trademark. And orange TM, as you can see, TM means the protection has been applied for. The trademark application form has been given for registration and yet not granted. So it is written as TM. And once you get a grant on that trademark, your trademarks get registered by the authority, concerned authority, then you get the R mark on that, that is registered trademark. And third is SM, that is the service mark, which is used for the services like banking service, courier service, transportation service, telecommunication. So all these things use SM, that is the service mark. And these are the other trademarks that I discussed, that is certification mark. As you can see, the FSSAI, the ICI, then the eco mark, eco-friendly products have a green pot-like thing, that is for the earthen pot, that is eco-friendly. So this mark is given to the products originating from the natural sources. Then the BSI mark, wool mark. So all these things are the certification marks, which are the names, symbols, and devices which are grouped by a person, showing the set of standards. Because if someone certifies, if a government authority or a concerned authority is certifying about the quality of that product, then the customer has the satisfaction of buying an original product which is safe for use. So it provides guarantee to the consumers that the product and services have desired standards. And other marks include the collective marks, which is formed by the member of collection of people owned by collectives to identify their goods and services, to distinguish from the non-member things, like the CA mark, that is the Chartered Accountant Services mark. This mark is for the members of the Institute of Chartered Accountants. Then service marks like FedEx is there for the courier services, which distinguishes it from the other services or business people in that area. Now, next form of IP that is of also very much interest, and that would be seen in all the areas, right from the beginning of our day with the toothbrush till the end of our day, that is at night with the blanket or whatever things that the all out that we will be using at night. So all these things are protected under industrial designs as well, with specific features like ornamental or aesthetic aspects of that article, which will be applied to any 2D or 3D features for with the shapes, colors, patterns, and line schemes. So this why this industrial protection is required. This patents are for technical aspects, the trademarks are for trade related aspects, and industrial design is for the appeal, for attraction, to make the product more appealing to the customer, because it would add value if you are giving very attractive, pleasant looking product. And along with that, it also increases marketability of product, but it does not protect any technical features. They would be protected only under patents. Only the aesthetic things could be protected under industrial designs. And for qualifying as a industrial design, it should also be new and original, and it should not have been registered in India previously and not previously been published. So these are the basic requirements to get a industrial design registration. And internationally it is governed by Hague Agreement and Locarno Agreement, and in India it is governed by Design Act and Rules 2001. And in other countries there are certain unregistered trademarks as well, like in some European countries and UK. And in India it is registered designs. It is considered once it gets registered, it is called as registered design. And in US it is also called as the registered design patents. These are not called design registrations, but are called as design patents. And that is also protected for 10 years, which could be further renewed for 5 years more. And so they can also be protected under copyright as an art of work. If you are designing something and you want to protect it under copyright, that could also be done. Two types of protection could be sought from this. And as you can see, it will be included for pharma as well as non-pharma, the commodities, the FMCG products. As you can see on the right hand side, right hand side of this, the various articles, various products that come under this category would be all forms of household goods. It may be automobile, it may be a mobile handset, it may be jewelry, apparels, then the medicines, the capsules, the design of capsule, the color scheme that has been used, the inhalers, other medical devices, injectors, nowadays auto injectors and others. They are also having protection under the industrial design. For the technical feature, they are protected under patents, and for the design aspect, they could be protected under industrial designs. The another form of IP, that is geographical indication, that is it indicates the quality of that or the what I would say is characteristic of a particular product based on the geographical origin. So an indication that originates from a definite geographical area, which is used to identify natural or manufactured goods. So it includes, the examples have been given here, like the Gadwal or Pochampalli saris, Nirmala painting, then Kolhapuri chappal. So all these things, there are around 450, not exactly, but around 450 products have been registered in India under geographical indications, as it indicates the peculiar qualities of that particular product are due to the origin, that is the geographical origin. Like Assam tea, the flavor aroma of that tea is because of the climatic conditions of Assam, the soil of Assam, the way it has been cultivated in Assam. So all these things together will give that particular qualities to that product. So that's why it is registered under geographical indications. And like Nagpur orange, they've got Alfonso mango. So these are all the, like Tirupati laddu is also being registered because the one that is made there, that is having unique taste and appealing aroma and all other things. So these things are considered under geographical indications. And internationally it is protected under Lisbon Agreement and under the appellation of origin. And in India it is protected under the Act 1999, geographical indication of goods, and the rules 2002. It is also valid for 10 years and could be renewed further. As and it will also give value, it will also increase the marketability of these products as they are originating from that geography. So the customer will tally the products from where it has been obtained, the source, and this will also encourage the people from that area where this has been grown to further market their product and to get certain commercial advantage over that. And it would also avoid others from selling the same thing with the wrong name. Like Darjeeling tea, if someone is cultivating it in, say, Maharashtra and selling it, saying it is Darjeeling tea, then that is not allowed. Certain legal actions could be taken against the person who are doing such wrongful acts. Next is the layout designs, that is the topography of integrated circuits. As you all know, integrated circuit includes various chips, transistors, resistors, microchips that are arranged on a semiconductor material plate. And all these diodes and all these things, the way they have been arranged on that particular plate or a device could be unique to that particular device. Like if you are having, you see different mobiles are there in market from different companies. So each mobile is having unique way of arranging these small transistors and devices so that they can make their mobile light in weight, and also having maximum features to be incorporated in a smaller device. So it will make the ever small digital devices possible. As you know, the smart watches are there with multiple features. So all these things are possible because of the unique way of arranging these circuitries. And this is done by the semiconductor SiC layout device protection, the layout in which they have been laid on the surface. And the term for protection is 10 years from the date of filing. And it is also governed in India under the Semiconductor Integrated Circuit Layout Act. As on the right hand side, you can see the different ways in which the circuits could be designed, and this could be protected so that others cannot copy the same way of designing and configuring the layout designs for their advantage without your permission. Trade secret is another very important and interesting form of IP, which is mainly applicable for the confidential information, confidential formulas, that could not be protected, or I would say that could be protected under patents, but are preferred to be protected under trade secrets. Why? Because if you are having some formula, that is, if you are making a formulation for pharma industry, a medicinal formula, so that you can protect under patent. But if you are making a spice formula of edibles material, or you are making certain sauce, you are making certain soup or beverage, and you are disclosing that formula, then anyone in their household or any small companies, any other companies can copy that formula and make that thing. But in other areas, like in the technical inventions, this copying could not be done. They will not be manufacturing those drugs and other things at household or in small areas. But this could be the better way of protecting such type of things. Like the spices, Coca-Cola formula is still protected under trade secret. Then the secret, what I would say, WD-40, that is one of the type of adhesives that is also protected. This is just one example. There are several other examples. Google algorithm is protected under trade secret. So many type of areas are there. Even the customers list could be protected under trade secret. Advertising strategies, business models, that could be protected under trade secret. The only thing is there is not a specific law in our country to protect, like trade secret law is not there, but these are controlled by contract law. That is the employees agreement, the contract between the employer and employee. The same 1872 law is applicable here, with certain arrangements, certain agreements that are being signed by the people who are working on the formulas that are protected under trade secret. And as the name indicates, it will remain protected until it is kept confidential and secret. So it is the protection is for perpetual duration, endless duration, and if you are breaking that confidentiality, then that protection is lost. Then anyone else can use that for their commercial advantage. Another form of IP that is that talks mainly about the literary and artistic work is the copyright and related rights. Related rights are also called as neighboring rights. So copyright protects the books, the paintings, the musical compositions, the artistic works, performances, broadcasting, playing. All these things are considered as the copyright protectable subject matter. And the owner is assigned, the owner also has the moral right for the work, that is the book or some performance or singing or composition or something. So they have the moral right, and along with that, any person can be assigned for a copyright for the creation that he has done or she has done. and it incentivizes and recognizes the person for that. One along with that, one provision is there, as you can see, for patents, industrial designs and trademarks. If someone is infringing that, you can stop them, you can sue them in court. In case of copyright, there is a fair use provision which allows the educational institutes, researchers, research and development people to use this copyrighted protected articles, the journal articles, research papers, for educational purpose, for teaching purpose. So that is considered as the fair use, which is not allowed in case of other intellectual property rights, but it is applicable in case of copyrights. In pharma, it is there for if you are doing some clinical trial or certain work for regulatory, then that patent protected invention can be used for studying those things for research purpose, but not for commercial exploitation. And the Copyright Acts are governed internationally by Berne Convention, and in India it is Copyright Act 1957 and Rules 58. So these are all the areas where the copyright protection could be sought, like cinematographic films, dramas, musical works, compositions, books, then drawings, paintings, layout designs, plant layout designs in industries, then the catalogs, newsletters, so leaflets that come with some medicine or some any of the products, softwares. So the software, one thing in India is softwares are not protected under patents, they are protected under copyright. If they are associated with some hardware, then only patent protection is sought in India for software patent, software invention. But in other countries, many countries, software inventions are also patentable. In India, software inventions are not patentable, but they are to be associated with some hardware to show some effect in hardware, then only it complies for patentability. Then the website content and all other things, which gives the expression, protection for expression and not for the idea itself. It is how you are expressing that in a creative manner. And the neighboring rights or the related rights associated with copyright include the broadcasting right, the rebroadcasting right, performance right. So, and the lifetime for any copyright protected book, say for Chetan Bhagat's book, so lifetime of the author plus 60 years. And it is from the date, that is 60 years from the date of publication is the protection term for copyrights. And the broadcasting life of the live performances, that is duration is 50 years, and rebroadcasting is 25 years. That is, you are rebroadcasting it via another media. This another form of IP is the plant varieties and farmers right, that is mainly inclined towards the agricultural area. That means the plant varieties which are previously defined, precisely defined by a group of plants. Means if a farmer is working on some plant varieties and trying to make some genetic modification or crossbreeding or hybrid varieties he is developing, then that varieties and the breeds, the breeders, new breeds that evolve from that could be protected under plant varieties and farmers right. So the person who breeds plants and has discovered and developed a new plant variety, that considers the breeder's protection for a new plant variety, applying for the grant of registration under this act. And the objectives for this is to protect the rights of the farmer, because that farmer can save, resow that, sale or exchange that new plant varieties. Then it protects the farmer's breeder's right for production, selling, marketing, and distribution, facilitates the growth of seed industry, because as the new plant variety is developed, that would be having some advantageous features like stability from climatic conditions, reproducibility with the same properties in the next crop, next cycle of crop. So all these things, new advantages, new features are developed in that new plant variety. So that right should be given to that farmer. So to give that right and to facilitate the growth of seed industry by getting more, what I would say, the fertile seeds that would be grown at a large scale, yield would be improved, safety would be there, stability would be there. So these rights are given by the Protection of Plant Varieties and Farmers Right, that is PPVFR 2000 one. And these are the main features for registration to be allowed, like plant varieties, breeders, extant varieties, farmers varieties, and all the elaborate farmers right. So to comply with these type of registrations, it should have different properties like novelty, distinctiveness, uniformity, and stability. So based on the compliance for all these requirements, the different rights could be given for the different plant varieties. Now, as here we are discussing about the pharma background, pharmaceutical research. And now you have a brief idea about what are the different forms of IP. Now we would see how these different forms of IP could be exploited to a maximum by the pharmaceutical industry and by the researchers. So here the patents, which are considered as the technical part, technical protection, protection to the technical inventions, to the scientific inventions. So that patents is the most important form of IP that could be used by pharmaceutical industry for protection of their inventions in this category. That is, they offer exclusive right to market their drugs and prevent others from selling, copying, manufacturing these drugs for 20 years from the date of application or date of filing. And the trademark, as you know, from different pharma companies have their own trademark, like GSK is there, Novartis is there, Pfizer is there. They are the brands, they have the brand power, brand value, and these help distinguish their products and protect their brand from on the company and the product level. Because based on the trust on that brand, the customer gets attracted or get inclined to buy the product from the pharma product from that company. And the prerequisites for the identifying, planning, commercialization, and protection of the inventions is the prerequisite for IP. Means if you want to identify, you have to plan, research, and develop, and then commercialize your product. So all forms of IP play an important role in that, for identification as well as towards the final commercialization of product, because technology would be involved, technical features would be involved, methods would be involved. So all these methods, technologies, the planning of production, how that would be produced, how the commercialization would be done. So these things could be judged based on the intellectual property rights that could be sought for these. And it gives incentives to the innovating companies, as they have invested a lot in research and development. That is called return on investment. So if you are investing your time, effort, money on all the inventions, conducting all the R&Ds, right from the identification of the active molecule till the final development of that molecule in a final formulation for a particular drug indication of a particular disease. So all these things need certain money, time, and efforts. So this effort should be incentivized. So patents provide such type of incentive to the R&D investment, R&D efforts. And with this, as more and more R&D is being done, new innovative drugs, medicines, vaccines are evolving, and this encourages the holistic development of the IPR industry in various disease areas. As you know and have experienced that nowadays, every now and then a new strain, a new virus, a new bacteria, a new form of disease is evolving due to the changes in lifestyle, due to the changes in the food that is there, due to pollution and other things. So for all these diseases, with R&D, new and new medicines are being originated, and they need to be protected under patents, so that further development is possible. And it allows the pharmaceutical companies to also have a strict action against counterfeit drugs. It also helps them to enforce their IP by filing some infringement suits in case someone is using them without their authorization. And it is also very, very important in case of mergers and acquisitions to target certain small enterprises, small companies, small SMEs, and from them the big companies can target this. And if they feel that the small companies are not able to manufacture, they have an innovative drug, they have an invention that is having a promising market but not able to develop that, then there could be chances of mergers and acquisitions with the giant points so that they could provide support for the development of such medicines, and that medicines could be available to the public for treatment purpose. And there are other chances like licensing, cross licensing, joint ventures are also there based on the capability of one. Suppose one company is having capability in one type of manufacturing facilities are there, the other company is having the innovative molecule, so that licensing could be there, that they could license their patented innovative molecule technology to the manufacturing company, so they can manufacture it, and there would be some licensing agreements between them. And this is the most important area for pharmaceutical industry. Maximum number of patents are filed in the area of pharmaceutical industry, related to drugs, related to medical devices, related to various combinations that would be giving additional advantageous effects. The various medical other than medical devices, there are other tools, other things that could be protected under the patents as well as trademarks and industrial designs. So here I have just captured a few images from Google Images, courtesy to Google Images, where you can see in the COVID situation, the subject matter that could have been protected under patents include various vaccines, like the examples have been captured here by Pfizer, Sputnik, Moderna. So they all had their own technologies. All these vaccines have been protected under patents. And as you can see the images at the end of the masks that are there are different grades of masks which are available: surgical mask, N95 mask, then self-containment mask, then P100 mask, respiratory mask. So all these masks could be protected for their technology under the patents. They could be protected under by trademarks by the company who is manufacturing them with certain logo, and they are also protected under the design for the distinctive design feature that they are providing, the way that that has been designed, the appearance of that mask, so that multiple protection could be obtained for a particular product. Similarly, there are certain treatment protocols, dosing. So if the particular protocol is followed, certain dosing is followed, that could also be protected under patent. Certain alternative packagings are available, delivery mechanisms have been developed, then certain diagnostic tools, RT-PCR test and other tests have been conducted in the pandemic, like for COVID pandemic. In that case, all the new technologies, even the electronic gadgets and other things evolved, which took care of the health of the human aspect, as well as they also had protection because of their R&D investment. So return on investment was there, but there should be balance between IPR and the human benefit, that is the public benefit. The medicine should not be too costly because to incur the R&D benefits, to fetch the R&D benefit. If it is too costly, then it would the layman could not have that. So there are positives and negatives for both IP protection as well as public health. So there should be a balance between these two, so that both of them get benefit, and new products evolve in market, and there should be some competitive advantage in market, competitive gain for the consumer to select the best thing. This is one of the case. Now I'll just brief with the Plexicon case, which talks about this is a case that is related to Plexicon patent infringement case with Novartis. So Novartis and Plexicon, they both were working in the same area, like for the cancer, skin cancer. BRAF is an BRAF inhibitors are the inhibitors. BRAF kinase are used for mutant melanomas for skin cancer. So this case is about Plexicon, which is a member of Daiichi Sankyo since 2011, and another party is Novartis. So Plexicon is a leader in the structure guided delivery and development of novel small molecule pharmaceuticals. And they have filed for 2 patents, US patents, which are considered here in this patent infringement case. I have included this case to let you know the value of IP. the weightage of IP, the enforceability of IP, and why it is important to protect your invention with intellectual property rights like patents, and how you can enforce your rights, how you can control the unauthorized use of your property. So the patents in question here are US 9640 and US 4539. I'm just reading the last four digits of that US patents. And these two patents cover Zelboraf, that is a drug trademark of that trade name of that drug, that is Zelboraf, and Turalio. These are the two drugs which got FDA approval in 2011 and 2019 respectively. And Novartis, which acquired GlaxoSmithKline in 2015, also traded in the melanoma drug therapy. So means Glaxo is active in various multiple disease areas. So the melanoma therapy division was taken over by Novartis in 2015. And later they started with the Tafilnar, that is developed by Glaxo in 2008. As now they have it has been acquired by Novartis. So this Tafilnar that has been patented by Glaxo is also acquired by Novartis, is also a property of Novartis now. So in this lawsuit, Plexicon alleged Novartis owed that they are using, means they are infringing the two patents, US patents that are described, given in the slides. So in this lawsuit, Plexicon alleged that Novartis owned the company's substantial royalty on the drugs Tafinlar, which infringes two Plexicon patents. Now the district court checked on this, and the jury trial at US district court was there, and Novartis conceded infringement. They said that no, this is not the case, we are not infringing your US patents, and they sought to invalidate Plexicon's patents covering Tafinlar, that is the two patents. They tried to invalidate that these are that these are already there on the grounds of anticipation, return description, and enablement. So they just took 2-3 grounds to invalidate the patents of the Plexicon family, that is Daiichi Sankyo family. So then Plexicon argued that a decade ago, that is more than 10 years ago, GSK was having a talk with Plexicon over certain partnership. So during that time, the GSK scientists who were involved in that description, in the meeting, in the discussion, partnership talk, they have seen the molecules, they have seen the new molecules, the structure of the molecule, core molecule, and then this partnership did never happen. But that scientist at GSK, they were knowing about this molecule. So evidence came in at the trial that GSK inventor who looked at the Plexicon's patent application disclosing the core molecule before shifting the program, focus in the way that ultimately led to infringement. This is jury's decision. As the GSK scientists have seen this molecule, they developed the same molecule in their laboratory, and subsequently as it is acquired by Novartis, then Novartis used this molecule for development of Tafinlar. So based on this thing, Novartis has infringed Plexicon's patent. So the jury rejected Novartis' argument and upheld validity of Plexicon's patent, and Novartis was found to be willfully infringing the Plexicon's patent. So Plexicon was awarded a damage of $178 million for the past infringement, and they will also be receiving royalties for the future sales of Tafinlar. So this is a power of patent, the power of enforceability of patent, how a patent should be monitored, enforced, so that others will not have a free ride on your invention for their commercial benefit. So this is how the patent could build value to a company. So you have to be very diligent when you are filing for a patent, whether you are working on that patent properly, whether it is being infringed by other companies, or you are infringing on others' patents. So all these aspects should be kept in mind while deciding to file for a patent, working on certain inventions to avoid any reinventing of wheel, and to get the proper protection on your intellectual property. So this is a very interesting case. So I have included this to get you and let you know about what is the value of a patent. Similarly, there are several other cases related to trademark infringement, copyright infringement, which could also be of very much interest. If you are interested in reading, you can go through certain IP sites to get an overview of such type of infringements. And with this, I would like to stop here and available for any discussion or the question answers or anything. Thank you for patient listening.
Thank you, ma'am. It was really a very informative talk. Ma'am has discussed about intellectual property rights for patents, trademarks, industrial design, and geographical indications. Ma'am has very well highlighted trademark secrets, copyrights, and related rights. Ma'am has also explained plant varieties and farmer rights, as well as the IP for the pharmaceutical industry and research. You have also highlighted some patent infringement cases, ma'am. That was very good examples. Your lecture was quite simple as well as eye-opener for everybody. So now the session is open for the queries. If anyone is having any kind of query, he or she can post their query in the chat box. We will unmute you. Anyone from the audience? Yes, sir. Good, sir. Sir, you can unmute yourself. You are co-host.
Yeah. Am I audible?
Anji, sir, you are audible.
I have in my lifetime heard many lectures by different authorities on IPR and patenting. But this is the first time I have listened to a lecture delivered with so much of information and with complete authority that Madam has delivered it, and it is a matter of pride for all of us that it has come from the mouth of a pharmacy professional. It is an excellent lecture and delivered in a very confident way. In this regard, I would like to congratulate Dr. Satvinder for making this lecture of eminent and very good speakers. This is about it. And now my own views on patenting and IPR issues are whether the learned speaker will agree or not is that IPR and patenting are valid and very good when there is a business concern, right? When there is a commercial angle,
Yes.
or it is benefited for industries, like you have made so many examples, like anyone who makes a vehicle engine which consumes half the quantity of petrol than the normal vehicle, it is good. It can be patented. But I am not very much agreeable in academic scientific pursuit in science, because science is universal. Science cannot be individualized. When a scientist makes a discovery and patents it, the science becomes individualized. The purpose of science is its propagation. Scientist A has an idea, he publishes. Scientist B improves over the idea, and scientist C makes applicable to that idea.
Yes.
But if you patent it and keep it to yourself, it's not a healthy trend for advancement of science. Now, in this regard, I ask you if Albert Einstein had patented his theory of E is equal to mc square, do you think the nuclear science could have developed so much? This is my view, and I don't know whether you share my view or not. This is how it. So, for the propagation of science, it is imperative that it should not be individualized. And I congratulate you, Miss, for your excellent presentation.
Thank you, sir. Yeah, actually, here I would just like to give a note that there are certain subjects which are considered as non-patentable subject matter that comes under Section 3 of Indian Patent Act. So the abstract theories, scientific principles, all these things are not patentable. No one can have monopoly on these things. So these things, if using these principles, scientific principles and laws applicable to a certain invention, that invention would be patentable, but these scientific principles are not at all patentable. So this is given with all examples and elaborations in Indian Patent Act in Section 3. Where Section 3D is also very good for very important and very unique in Indian pharmaceutical industry. Section 3D that talks about a new form of a known substance like ester, polymorph, other derivatives of a known chemical substance is not patentable unless and until it is giving a certain advanced substantial therapeutic efficacy, enhancement of therapeutic efficacy, advancement of efficacy. Like in other countries, there is patent evergreening because the new forms of the drug, like the new ester, new salt, they get patent on that. But in India, being a developing country, we do not want to have all the patent implications on all type of drug discoveries. So here, that this India is considered as the taking its own stand over that these are not patentable. So Section 3D, which is unique, and we are proud to have this section. Otherwise, in India, it would be very difficult for the common man, the public, to get the drugs on a reasonable at a reasonable, affordable cost.
Thank you, madam, and my personal compliments to you for such a nice delivery.
Thank you, sir. Thank you. My pleasure.
Thank you, ma'am. Thank you. Now it's time to felicitate ma'am Kavita Kanoje. Kindly accept our token of gratitude, ma'am.
Yeah.
Thank you.
Thank you, ma'am.
Thank you. Thank you, sir. Now I would like to invite Dr. Sonia Singla for the vote of thanks. Please, ma'am.
Honorable Principal, Dr. Satvinder Kaur, respected staff members, and our most valued invited guests and resource persons, our dear delegates, and it's my privilege to have been asked to propose a vote of thanks on this 8th day of faculty development program. I, Dr. Sonia, on behalf of GHG Khalsa College of Pharmacy and the entire management, APTI Women Forum Punjab State, even special guest, and on my own behalf, extend a very hearty vote of thanks to all the speakers for gracing your important work and sharing with us your findings and opinions today. A big thanks to Dr. Monica Gulati for their motivations, inspirations, and deliberations. I must mention our deep sense of appreciation for Professor Vandana for her explanation about drug-eluting stent market and stent customization and vascular interventions. Further, we are grateful to Dr. Manjinder Singh Gill for giving an excellent coverage to diphenyl-mediated carbamate synthesis with their optimized studies, synthesis of sulfonyl ureas and carbamate-based different compound synthesis. I may also like to express our sincere thanks to Ms. Kavita ma'am for demonstrating intellectual property rights, trademark, trade secret, copyright, and related rights. We are all inspired by you, by your great words. Thank you all. We will meet virtually again tomorrow on 9:50 a.m. sharp. Schedule for the day will be shared with you in WhatsApp group. Again, thanks to all. Over to you, Anvinda ma'am.
Thank you, ma'am. Thank you. We will meet again at 10:45 on the 9th day of FDP. Thank you. Thank you all.
Thank you.